is going on? Can you guys hear me?
Yes.
Yes.
Can someone say something so I can see if I can hear you?
Hi, can you hear me?
Can you hear me?
Well, that is peculiar. I am not able to hear anything. What is going on? How about now? Can someone say something now?
Can you hear me?
Okay. Now it's working. All right. I'm not sure what happened there. I picked a weird speaker that I didn't have. All right, cool. I can hear you. You can hear me. We are on our way. Excellent. All right. Welcome. Today is Monday, March 15th. We are into our digestive system material. This is just a quick and easy section. That's how I like my sections. So it's going to be a quick 3 lectures, and then we are on to the exam and then on to spring break. So it's going to be a nice, and again, pun is included, intended, palate cleanser for what we kind of the heavy stuff we were doing in the past section. We are going to focus on our digestive anatomy and the system, working our way through that today. You do have 3 assignments for this section, and that is going to be 1 review, the Unit 24 review, 1 Physio X, Exercise 8, which has 4 activities, and 1 Lapster, which is the intestinal glucose transport, which again, for full credit, you must complete 100% and also get at least 80% correct. And again, I know in the past, remember, you can do these Lapsters more than 1 time. So if you don't complete it 80% correct, you won't get full credit, but you can always go back and do it again to get 80% if you want to get that full credit for that. So you may complete it as many times as you want, and it will save your highest score. All leading up till Wednesday the 24th, when we have our lab and lecture exam. Then you get a whole week off for spring break. And so when we come back, the pre-labs for our urinary system and respiratory system, which we'll be doing in the next section, are going to be due when we come back from that. And that's it. It's a very, very simple, straightforward schedule. So any questions on that? I am knee deep in your exams. I hope to have them done. I expect to have them done, knock on wood, by Wednesday. So unless something surprising happens between now and then, I just freaked out my dog by knocking on the desk. I should have them done by Wednesday and have them back to you. So that should be good as that. So we'll talk about that more when we get to that. All right. You don't want to know. In fairness, I started with the lab exams grading because they're harder to grade, and they are a diverse set of scores. Let's say it that way. But I'm guessing the lecture exams will be better. I'm hoping the lecture exams will be better. And I haven't curved them yet either, so hopefully the curve will help as well. All righty, let's go ahead and dive into our digestive system. Like all organ systems, as we've talked about, we have 2 main groups of organs. Our primary organs of the digestive system, what are commonly known as the alimentary canal or your gastrointestinal tract. And as we've talked about, this is a tube that basically goes from your mouth to your anus that in a cadaver is about 30 feet long. Now, notice I had to add that caveat there, in a cadaver. Don't you guys all have tubes that go from your mouth to an anus? Yeah, but in you and in me, it is not 30 feet long. Why is that? Is it longer or is it shorter?
The stress makes things shorter.
It is shorter, and absolutely. Why is it shorter?
Muscle tension.
Yeah, absolutely, right? Death is many things, but one of the things it is very, very relaxing. Our digestive system has a tremendous amount of muscle tension in it. And so when that muscle tension relaxes, it does expand to its full 30 feet in length. But basically, this is that hollow tube that we've talked about that our food passes through. And remember, as we also talked about before, and I'll remind you again when we were talking about exocrine glands, the lumen of our digestive system is outside of the body. Right. So again, remember, we had this fancy term for it where we referred to it as the topological exterior. it makes the digestive system somewhat unique in that basically most of the processing that takes place in it takes place outside of the body. So most of the processes, most of the things that occur outside of the body. And then, of course, the ultimate goal of that is then to prepare the food so that it can cross that membrane into our body. And that's also how we pretty much identify the organs of the alimentary canal. These are the organs that form the hollow tube and that food passes within. So give me some examples of some of the organs that would make up the alimentary canal. Stomach, esophagus, yep. Mouth, small intestine, exactly, and so on and so forth. Excellent. Perfect, perfect, perfect. Like we know, most of the organs of the primary organs are what are responsible for the primary functions. However, we do have a series of accessory organs that help to assist in that process, right? They assist in those major functions of the digestive system. And give me some examples of some of the accessory organs of the digestive system. Gallbladder, pancreas, teeth, liver, exactly, right? And again, a good way to think about whether it is an accessory organ or whether it is a primary organ is does food pass, within it, right? Again, and it gets a little tricky with teeth and tongue, right? But again, unless you've got a really large piercing, food doesn't typically pass through your tongue. It may go over your tongue, it may go between your teeth, but again, unless you have a really large cavity, it's not going through your teeth, right? Food doesn't pass through the liver, food doesn't pass through the gallbladder, and so on and so forth. So it's a pretty easy way to tell what is what. All right, and as we mentioned, those primary organs are responsible for the primary functions, and there are 6 primary functions of the digestive system. So give me an example of what one of those might be. What are some of the functions of the digestive system?
Catabolysis of macromolecules.
Okay, excellent. I like that. What do you mean by that?
Breaking down of complex molecules into simpler constituent molecules that can be used in anabolic processes.
Right, absolutely. The breaking down of food. And you are absolutely correct. However, we want to be careful when we talk about breaking down of food because there's really 2 types. There's the catabolic one that you're talking about. And when we break something down catabolically, what are we really breaking?
Chemical bonds.
Chemical bonds, exactly. So we have a chemical, what we call a chemical digestion. And that chemical digestion is when we are breaking the bonds that break, polysaccharides into monosaccharides, that break proteins into amino acids, right? That break lipids into fatty acid chains and alcohols, things along those lines. So we have a chemical digestion where we're catabolically breaking those bonds. And of course, typically we are going to use enzymes to be able to do that. But we also have a mechanical digestion. This mechanical digestion is just the physical breaking of big things into little things. Can you give me an example of that type of digestion?
Chewing.
Mastication.
Yeah, mastication, absolutely. Right. I'm sure everyone in this class, when they take a bite of food, chews their food the 20 times that you're supposed to chew it before you swallow it down. Right. And that process of mastication is where we are taking those big pieces of cheeseburger and breaking it down into small pieces of cheeseburger, but we're not changing the composition of it. It's like taking a big boulder and a hammer and a chisel and taking that big rock and breaking it down into a bunch of little rocks. We've changed the surface area, we've changed the shape of it, but we haven't changed the composition of what it is. right? Chemical digestion is where we're breaking those bonds, and we'd be separating the zinc from the, you know, the metals and all the other things that would be in that rock. Whereas the mechanical digestion is obviously where we're breaking big things down into little things. And really, the main reason we do this is to increase surface area. And when we increase that surface area, that makes it easier for those enzymes, to do their job. Excellent. All right. Give me another example of a function of the digestive system.
Move it.
Movement, absolutely. Movement. There's all sorts of different types of propulsion that are going to take place from voluntary acts, or at least semi-conscious voluntary reflexes involving swallowing, deglutition, then also all sorts of propulsion. Again, all of these 6 processes of the digestive function don't occur in just one organ in one location. So pretty much every organ of the alimentary canal plays a role in moving that substance from one location to another. And there's lots of different types of propulsion processes we will talk about. What else?
Excrete.
Say again. Well, okay, I see where you're going there. Absolutely. We do have glandular secretion, although I would argue that you could say that a glandular secretion.
No, I said excretion.
Okay, well, so you mean, I'm not sure what you mean by excretion then.
I mean, like waste, like moving waste out of the body.
Oh, okay. Oh, defecation. I see what you're saying. Absolutely. Defecation. Defecation. All right. While we are able to absorb most of the components of the food we ingest, there is the removal of undigestible material and other stuff out of the alimentary canal. Excellent. I misunderstood what you said before, but it is a good point too. When we think of secretion, that secretion is typically components, enzymes, like we talked about for the chemical digestion, or things that help to facilitate the mechanical digestion. So we could argue that secretions that are released from our alimentary canal primarily help these 2 processes. So you can kind of fold them into that. All right, so we got our breaking down, our digestion of the food, our momentum of the food, our defecation of the food. What else?
Ingestion.
Nutrients.
Okay, excellent. I like that. So absolutely. So perfect. Those are both good ones. Excellent. We have the absorption of the materials. Oops. The resources that we need. Again, the whole point of breaking things down chemically and mechanically is so that we can cross that membrane and enter into the body the things that we want and the things that we need. And I think I also heard someone say ingestion, which is, again, one of those voluntary acts of the placement of food into the alimentary canal. Oops. Excellent. So we have our chemical digestion, our mechanical digestion, our movement, our defecation, our absorption, and our ingestion. Excellent. I think we have all of them. And I like the order that we came up with this with, because unlike some of the other organ systems we've talked about, we have these lists, and here's our list here. Obviously, some of these events have to occur before others. Ingestion has to occur first. Defecation typically occurs last, right? But does that how it works? We ingest the food, and then we do all of the propulsion, and then we do all of the mechanical digestion, and then we do all of the chemical digestion, and then we do all of the absorption?
No.
Of course not. Most organs in the alimentary canal are responsible for one or more Of course, keyword in that sentence is most. There are a couple organs that are primarily just involved in one, but most of the organs in the alimentary canal are responsible for more than one of these processes. Can I go back to the whiteboard? Sure. Just has the schedule in it, but I'm happy to do that. I think it just has the schedule in it, right? There you go.
Oh, I actually meant the screen where you had the notes. I apologize.
Oh. All right.
So I can take
Yep.
a picture.
Hold on, I need to. Well, again, remember, you don't have to take a picture. You can always go back to the lectures and watch them again. But let's do that. Actually, you know what? Because I changed the screen, unfortunately, I've lost that. So.
Oh, okay. Okay, no worries. Thank you.
But again, here are the 6 processes. We just
Yeah.
did a little more writing on it. So I apologize for that.
Oh, no worries. Thank you.
I switch screens off of the slides. The things that I put on the slides don't stay. The things on the whiteboard stay, but the things that I put on the slides don't. So I apologize for that.
Okay, got it. Thank you.
All right. And I guess really the only thing that we don't really have here that we talked about is you could argue that there is a 7th process, and that 7th process is secretion. Right, the release of substances. But as we mentioned, that those secretions that are produced and released outside of the body into the lumen of the alimentary canal are primarily serving either a mechanical or a chemical digestive function. So you could also, if you wanted to think of it as a 7th function, that is fine. If you want to fold it into these 2, that's fine as well. I don't care about the numbers as much as I care about that you guys have and understand everything that the digestive system does. All right. And of course, we have the pretty picture here emphasizing the point that we just made. That again, that is not a linear list of steps where 1 first and then 2 and then 3 and then 4. It doesn't work that way. As I said, most organs in the alimentary canal are responsible for 2 or more of these digestive functions. And here we have this nice picture from the textbook. It kind of shows, obviously, propulsion occurs throughout the entire system. Absorption occurs in multiple locations, chemical breakdown in multiple locations, mechanical breakdown in multiple locations, and so on and so forth. Now, one thing we are going to do differently about the digestive system than we've done from previous organ systems is primarily we've done all the anatomy first, and then we did all of the physiology. However, because of the length and the diversity of the organs in the alimentary canal, in this case, I think it makes a little bit more sense for us to work our way the same way that cheeseburger that you had for breakfast is going to work its way through the alimentary canal, following it along on its mystical and magical journey as it works its way from mouth to anus. So that is how we're primarily going to focus. We'll go organ by organ, working our way through the primary organs, talking about the accessory organs that are associated with them and the functions of all of those. However, that doesn't mean there aren't some basic concepts for the functions that occur throughout our alimentary canal. And so let's talk about that first. As we already mentioned, one of the unique characteristics about the digestive system is it regulates the environment outside the body, right? In the lumen of our alimentary canal, our gastrointestinal tract, which again, as we said, is the topological exterior of the body. And there are basically 2 main basic digestive processes, right? Our goal is to initiate some type of stimulus. Initiate the process with some type of stimulus, typically some type of chemical or mechanical type of stimuli. And as a result of those, we're going to get basically 2 main effects in our alimentary canal organs. We are either going to get an increase or decrease in glandular activity, producing more or less of a secretion. And we'll have a change in the smooth muscle activity affecting propulsion and other type of mechanical events that are taking place inside of that organ. Now, of course, these things aren't occurring randomly, so that means we need to control them. And what's the primary way we control things in the body, or primary ways, plural, that we control things in the body?
Neurologically and hormonally.
There you go. Using neurons, using hormones. Absolutely. There's going to be a neurological control and there's going to be a hormonal control. So let's talk about those. First, we will talk about the neural controls. And one of the cool things, we talked about this a little bit back in 430, and we'll talk about it a little bit more here, is with the neural mechanisms, there is this big elaborate plexus. Does anybody remember what a plexus is?
A collection of nervous tissue.
Yeah, I like that. It is an elaborate collection of typically nerves and ganglia, and this doesn't have to be this big anymore. So obviously this is in the peripheral nervous system. Oops, peripheral nervous system. An elaborate networks of nerves and ganglia. And someone remind me again what a ganglia is. All right, let's start easy. Someone remind me what a nerve is. Did my speaker go out again?
Nerve is specialized tissue that allows for the conduction of afferent and efferent signals.
Okay, I like that. It absolutely, nerves are indeed highly specialized for the production of action potential. What are nerves comprised of?
Neuron,
No, what part of the neuron?
the axons and dendrites.
Not and dendrites. You're getting closer there. Nerves are basically bundles of axons. Remember a nerve, and nerves come in two flavors. What are the two flavors of nerves again?
Motor.
Okay, you're right. Functionally, I wasn't thinking functionally. I was thinking structurally. Structurally, what are the two main types of nerves?
Bipolar and multipolar?
Well, again, they're axons. So how about nerves are either sensory or motor, right? Carrying information in or out. They're or anatomically, they are, so that was the functional, cranial nerves and spinal nerves. How many cranial nerves do you guys have again?
12.
12
pairs.
pairs, absolutely. How many pairs of spinal nerves do you have again?
31.
31, excellent, absolutely. So remember, a nerve is a bundle of axons, right? Whose job is to carry information from one location to another. So if a nerve is a bundle of axons, someone remind me what a ganglion, let's go, well, I guess I went, let's go this way. We'll go singular. Nerve and ganglion, we'll go singular. What is a ganglion? You guys hurt my heart a little bit with this.
The nervous system was a struggle.
Well, if a nerve is a bundle of axons, what does that make a ganglion a bundle of?
A bundle of nerves.
Nope, not a bundle of nerves. Are neurons just axons?
Get the feeling that we're on your last one.
Nah, it's Monday morning early. I'm doing good so far.
Bundle of axons.
Well, again, so neurons are not just made of axons. What are the other parts of neurons?
Dendrites.
And
Synapses.
okay, a synapse typically occurs between an axon and a dendrite, but there's also the largest part of the neuron, which is the
Cell body?
cell body. Absolutely. Remember, a ganglion is a bundle of cell bodies. And I guess you could argue and dendrites as well. All right, bundle of cell bodies in the peripheral nervous system. So again, we have these plexuses, which are elaborate networks of nerves and ganglion that serve some elaborate function. And one of the most elaborate plexes is the enteric plexus. It what is often affectionately referred to as the gut brain. Many of the basic decisions that take place in the digestive system are actually decided on by the digestive system. Essentially, this is responsible for very basic reflexes, what we call the short reflexes or intrinsic reflexes. These are intrinsic, and actually I'm going to cheat, and now I will go to the whiteboard. We don't need that, so that's fine if it goes away. So when we talk about our intrinsic control or our short reflexes, there are some key factors to them. The first is that they are local in their effect. Typically what this means is, again, it's a reflex. So if you think about it, with a reflex, we have a stimulus. Then we have a command center where we process that information. And then we have an effector. Being local in its effect, that typically means that the effector is the same organ where the stimulus occurs. So with the stimulus, it is a local stimulus inside of the organ. And the effector is typically the organ that was stimulated. So you have that nice big cheeseburger for breakfast. And when you have that cheeseburger for breakfast, your stomach stretches. And when your stomach stretches, the result of that is it is going to start churning up that food, and it is going to start releasing hydrochloric acid, releasing enzymes, releasing those things to break that food down. So notice we had a local stimulus and we had a local effect. Now, when it comes to the command center, is that something that you had to consciously decide? All right, stomach, here comes the cheeseburger, get to working. No, in this case, that command center, the decision-making process actually occurs in that enteric plexus. That's why it's called the gut brain, because many of the decisions that your digestive system make of how it's going to function occur right here inside of the alimentary canal itself. So when your stomach stretches, your stomach says, hey, I'm stretching. That means there's food inside of me. I need to churn. I need to make more gastric juices. And this is our intrinsic control.
Question.
Yes.
So as far as reflex arcs go, for an intrinsic control, does the reflex arc skip the central nervous system in favor of the enteric
Yes.
nervous system?
With these short reflexes, absolutely. With these short reflexes, it does not go to the central nervous system. That is absolutely correct. All right.
So then is there a, I don't know, like peripheral kind of, I'm not even sure how to phrase it. Is there something that from the enteric nervous system that jacks into the central nervous system in order for the central nervous system to regulate some of those processes?
Well, I'll answer that question two ways. Notice we didn't call this control and reflexes. We called it intrinsic control and short reflexes. What's the implication there?
It's all low.
Yeah, that there's going to be an extrinsic neural control. And actually, let's throw neural in there. Also helps if I spell intrinsic correctly. Intrinsic. There we go. And even if you hadn't thought about it that way, you're absolutely aware of it. Because, right, obviously, when you eat that cheeseburger becomes active. But if instead you're sitting on the couch watching a Carl's Jr. commercial, right? There is nothing that looks better than a Carl's Jr. cheeseburger on the commercial. I'm not saying they look like that in real life, but the commercials, you could be a vegetarian and your mouth is going to start watering when you see that Carl's Jr. cheeseburger. Your stomach's going to start rumbling when you see that Carl's Jr. cheeseburger. Clearly, the stimuli is coming from outside of the alimentary canal in that effect, but can it absolutely positively influence your alimentary canal? Absolutely. And that is the key to the second type of reflex. And you're absolutely correct about that. It is an intrinsic neural control, or what we call the long reflexes. Again, one of the keys to this long reflex is typically, so again, this one typically was local in their effect, and I guess I should sneak typically in there because again, you know, there's always exceptions. With our extrinsic control, typically can affect multiple organs. Absolutely, we absolutely do want. Oh, I see. Again, both terms are acceptable. So both, you can use long reflex, you can use short reflex, you can use intrinsic, you can use extrinsic. Both are acceptable ways of describing these. So I'm fine with either way. And there are some key differences. The first come in terms of the stimulus. In the stimulus for an extrinsic pathway, the stimulus can be inside or outside the alimentary canal. Obviously, if you are tasting that cheeseburger or when that cheeseburger hits your stomach, it has the ability to influence an extrinsic effect. But as we talked about, watching that Carl's Jr. commercial, or if instead your neighbor who loves to barbecue all the time has that tri-tip out on their barbecue, you know, that one that they've been marinated in the bottom of their fridge for 3 days, and then they brought up for room temperature and they put it on that really hot grill to get that nice sear on the outside, and then went slow and low to perfection, and you smell that wafting over the fence into your backyard. Can that influence a change in your digestive system? Yeah,
Starting to sound like culinary school.
exactly. And that's the point, right? It can be inside or outside, right? It can be smell, it can be sight, right? It can be, you know, taste, stretch, or it can even be mental. If you really, really think back to your last anniversary where you guys went to Ruth Chris for that amazing meal and you had that wonderful, you know, hickory smoked steak with the butter, herb butter melted on the top of it. And if you really just sat here and thought about that, could you start salivating, thinking back to all those things? Or again, and if you're vegetarian, you know, that roasted portobello mushroom or whatever it is that tickles your fancy. All right, absolutely. If you just sat here and thought about food, do you think you could affect your alimentary canal? Affect your digestive system? Absolutely. And so that's one of the keys to this. It can occur both inside and outside the alimentary canal. The reason for this is, as was mentioned, in this case, the command center for this is going to include our autonomic nervous system. Again, it's still not conscious control. I'm not thinking, hey, that cheeseburger looks really good, I should start salivating as a result of this. It's going to be our autonomic nervous system. And of course, this means both our sympathetic and parasympathetic. Now, when we think of this type of command, sympathetic is typically excitatory or inhibitory?
Excited.
Excitatory.
Sympathetic for our digestive system.
Inhibit.
Yeah, so typically our sympathetic is going to be inhibitory, and our parasympathetic is going to typically be excitatory, right? And again, a lot of that is personal preference. As I mentioned, if that neighbor happens to be bringing over a big plate of that perfectly cooked tri-tip that's nice and pink, and you can see all the bloody juices coming out of that, for many of you, that may excite your autonomic nervous system and excite your digestive system, unless, as I pointed out, you happen to be a vegetarian. Right? If you're a vegetarian, are you going to start salivating at that tri-tip that's being brought over? No. And again, what if while carrying that over, he trips and drops it and the tri-tip lands on your dog poop in your backyard? Is that going to change the way that you respond to it? Yeah, absolutely. Right? Or if you're a little kid being asked to eat broccoli, right? Most little kids would rather have their left arm ripped off than eat a sprig of broccoli, right? So obviously there are a lot of top-down processing that can occur in this. And again, we can get both excitatory and inhibitory inputs to influence and affect our digestive system. So this one does go through our central nervous system. And again, that makes sense. If you're seeing or smelling or even tasting, we know those go to the central nervous system, and then our autonomic nervous system, our sympathetic and parasympathetic pathways carry that information to our organs. And again, as we mentioned, typically to multiple organs. And that's really the key to the effectors. With the effectors, typically will affect multiple organs. So again, obviously taste can be a part of this extrinsic pathway, and tasting something really good can increase saliva in your mouth, but it can also affect what's happening in your stomach. It can also affect what's happening in your small intestine. It can also affect what's happening in your large intestine. And it may not be as obvious for you, but those of you who have kids know this very, very well. Because a kid sits down, you get that 2-year-old, 3-year-old who sits down at the 2 and 3 is probably the key gauge for this. They sit down at the dinner table, they have 2 bites of food, and they instantly jump up. Because where are they going?
Bathroom.
Yeah,
Play.
bathroom to poop. Exactly. They're going to the bathroom to poop, right? Absolutely. So again, it's because that stimulus of the food has affected multiple systems in the body and causes defecation, triggers defecation for them. So notice these are both neural controls, but there are some big differences between them. We've done it here with the words, and we've got it here as well. Again, our intrinsic controls are our short reflexes, regulated by our enteric plexus, mediated by local cues that are occurring inside the alimentary canal, and have a local effect. So intrinsic control of the stomach affects the stomach. Intrinsic control of the mouth affects the mouth. Intrinsic control of the large intestine affects the large intestine. Extrinsic controls are our long reflexes. They can occur both inside and outside the gastrointestinal tract. Sight, smell, hearing, right? Thinking, all of those things can influence it. It's still autonomic. It's not voluntary control. But those sympathetic and parasympathetic influence our enteric nervous system and affect our digestive function. And primarily, what nerves are involved in this? I like this. Let's actually go back to the whiteboard for this. In our extrinsic pathway, as we mentioned, there's sympathetic pathway, and we'll start you guys off easy. What is the parasympathetic pathway to the digestive system? I'm not giving you this one because this one you know. I can wait. I have grinder. Swipe right. Swipe right. Ooh, swipe left. Come on. What's the parasympathetic pathway to the digestive system?
It's a cranial nerve.
Yeah, which one? You got a 1 in 12 chance of getting it right. There we go. Vagus. So which cranial nerve was that again? 10. Cranial nerve 10. Absolutely. Excellent. The vagus nerve. Absolutely, right? Vagus nerve, 90% of our parasympathetic output, including to all of our digestive organs, right? In the sympathetic pathway, remember it was our prevertebral, or what we also call collateral ganglia that innervate our digestive system. So if you think about that in terms of nerves, you know, the organs below the diaphragm, those would be, remember, what we called our splanchnic nerves. So it is the splanchnic nerves. Yeah, it's vagus, absolutely. So the splanchnic nerves from those prevertebral or collateral ganglia are what innervate most of the organs of our digestive system for that. So again, we've done this all with the words, but here we have the pretty picture that kind of emphasizes this same thing. Notice again, when we look at our short reflex, right here with our short reflex, we have some type of stimulus that is occurring inside our alimentary canal, right? There's some type of change, a mechanical receptor like a stretch, or a change in the chemicals, or a change in the osmolarity of the lumen. That sends a signal to our gut brain, that enteric plexus, which then sends a signal to either our smooth muscle and glands, and we affect some change inside of the organ where we had the original stimulus. However, remember also that an internal or external stimulus can be processed by the central nervous system. So again, taste in the mouth, smelling, thinking about food, all of those things can be processed by the central nervous system. So your response to the taste of a mushroom and my response to the taste of a mushroom and the response to a 3-year-old tasting a mushroom, we're all getting the same internal stimulus, but how we respond to that is going to be very, very different, primarily because of this long reflex. You may be a vegetarian who loves mushrooms. That kid may think it tastes like dirt, right? So it's going to get processed there. Then our autonomic nervous system, both sympathetic or parasympathetic, carries those efferent impulses back to our effectors, to our gut brain, where we affect some change and we get some type of response. And again, with these afferent impulses, remember, they can go to multiple organs. So unlike the short reflex, with the long reflexes, we can affect typically multiple organs with that. So we've done it with the pretty words. Here we've done it with the pretty picture. And we have talked about the way we can neurally control our alimentary canal. All right, questions on that? All right, excellent. Of course, there is going to be hormonal control as well. Now, again, there are dozens of hormones that are involved in digestion. So I'm not just going to sit here and rattle them all off. Your book has a nice table that identifies them, but also as we work our way through the organs, we will talk about some of the hormones that are actually released by those organs, because most of them come from what we call enteroendocrine cells. Right? We've already talked about endocrine glands. However, in our alimentary canal, they're typically individual cells with lining the mucosa of our alimentary canal that we call enteroendocrine cells. And these are single-celled endocrine glands. So just within the wall of our alimentary canal, occasionally there will be a cell there, and that cell is capable of producing one of many hormones. Like I said, over a dozen different hormones that are produced by the alimentary canal. We are not going to learn all of them. We're going to focus on the key ones and the ones that I like. Like all hormones, they can be circulating hormones, which allows them to affect multiple organs, multiple parts of the body, and basically allows us to coordinate the activity of many of the structures in our alimentary canal. And obviously, being hormones, they can also affect, influence systems outside of our digestive system, right? And again, you may not have thought of it in those terms, but again, when you took your wife to that fancy dinner, or your significant other to that fancy dinner at Ruth Chris, and you decided to get that tomahawk chop, right? All 45 ounces of it, and you got about 3/3 of the way through it, right? You start getting those wonderful meat sweats, right? So is digestion affecting your integumentary system when that occurs? Yeah, absolutely. So we have that ability to affect other organ systems when these types of things occur as well. All right. So like I said, we'll talk more about individual hormones as we run into them in different organs, but it is an important control mechanism. So questions on that? Excellent. Love my stunned silences because that tells me that you guys have mastered the material and means I can make the tests harder. Excellent. All right. The other basic thing we need to talk about is the basic anatomy. Now, if you remember when we way back in 430 talked about the tissues and we talked about the alimentary canal, we know that the openings of the alimentary canal, right, the oral cavity, the pharynx, those, and then of course the anus, are lined with a very special type of epithelial tissue that allows them to be protected from the outside world. And what was that tissue that lines the openings to our alimentary canal again?
Non-keratinized stratified squamous.
Actually, a non-keratinized stratified squamous epithelial tissue. Excellent, excellent, excellent. As we mentioned, that covers the anus, it covers the oral cavity, the pharynx, and it covers the proximal 2/3 of the esophagus. So notice from the distal third to the rectum, oops, if I spell rectum right, our alimentary canal is lined with a different type of tissue. And what was that different type of tissue that lines our alimentary canal from the distal third of the rectum, distal third of the esophagus to the rectum?
Simple columnar.
Simple columnar. Excellent. So even way back in 430, this was a concept that we had talked about, that there is a difference between the very beginning and the very end of the alimentary canal, and the majority of it in the middle. All right. So basically 27 of those 30 feet basically have the same anatomy to it. But the ends are something different. And it turns out that isn't just true for the epithelial tissue. When we talk about the basic anatomy, and here I'm going to cheat and get rid of that and that and that and that and that. When we talk about the basic anatomy of the alimentary canal, not only is the epithelial tissue the same, but the same basic functional anatomy is in place for the majority of the alimentary canal. The distal third of the esophagus to the rectum basically has this basic anatomy that we see here. So when we talk about the microscopic anatomy of the alimentary canal, this is the basic anatomy for the majority of it. Now we have the pretty pictures of this here, but you guys know how much I love drawing things. So let's go ahead and go to our whiteboard and talk about these layers. For the basic anatomy of the alimentary canal, there are four basic layers, four main layers. The first layer, as I'm sure you are aware of, so again, we will go from lumen up here to the abdominal pelvic cavity. Cavity here. Our first layer lines the surface that is open to the outside world, because the lumen is the outside world. And of course, we know that there is a membrane that lines the organs that are open to the outside world, the cavities that are open to the outside world. So what type of membrane is going to line the lumen of our alimentary canal?
Mucosa?
Yeah, mucous membrane, absolutely. A mucous membrane, or as you, mucus obviously is the adjective that describes the membrane, or we can just simply say mucosa, which is the noun. Absolutely. And as you guys know, our mucous membrane, so this is our first layer, the mucous membrane. Mucous membranes, like all membranes, are comprised of an epithelial tissue that sits on top of a connective tissue. And I've already given it to you. What is the epithelial tissue that is going to line this?
Simple columnar.
Simple columnar. Excellent, epithelial tissue.
For the mucous membrane, was simple columnar?
For the majority of the alimentary canal. Now remember, in the mouth, the mucous membrane, in the anus, the mucous membrane is going to be that non-keratinized stratified squamous. But again, we're talking about the basic anatomy. So basically from the distal third of the esophagus all the way to the rectum, it is going to be simple columnar epithelial tissue. Absolutely. Now, what connective tissue is part of a mucous membrane?
Lamina propria?
Well, so
Areolar.
we do give this connective tissue a name based on its location. And that name based on its location is indeed the lamina propria. Absolutely. But what actual type of tissue is that lamina propria?
Areolar.
There you go. Areolar connective tissue. So let's cheat. If I open this up, can I do that? There we go. And then I can move this over here. And then I can actually draw it. So we'll go with purple. So here is my simple columnar epithelial tissue. Pretend these all are columns. You get the idea. Excellent. And we know it sits on top of an areolar connective tissue. However, there is something extra in this particular mucous membrane. In this mucous membrane, there is actually a third layer. And this third layer is the muscularis mucosa. With a name like muscularis mucosa, what do you think it is?
Smooth muscle?
Smooth muscle, absolutely. There is a thin layer of smooth muscle forming a third component to our mucous membrane. Obviously, as mentioned, this is indeed smooth muscle. It isn't something we voluntarily control. Why do you think it's here? Is a tiny thin layer of smooth muscle going to be enough to move that cheeseburger you've had from breakfast from your stomach into the small intestine?
No.
No, probably not.
No.
We're going to need a lot more muscle to do that work. So it's going to need a lot more than this tiny, tiny little layer that we see here, right? In fact, let's go back to the pretty picture from the textbook for a moment. Notice as we look at the pretty picture from the textbook, right? Again, here we see that simple columnar epithelial tissue, a little bit of areolar connective tissue that we call the lamina propria, and then this thin layer of smooth muscle. Notice there's much more smooth muscle to be seen later on. So this isn't necessarily going to be for propulsion or movement. So what is it going to do? Any idea what it might be useful for?
Changing the size of the lumen.
Yeah, it can change the shape of the lumen, change the surface of the lumen. That can increase and decrease surface area. Or, again, if a little piece of that, instead of a cheeseburger for breakfast, you had a peanut butter and jelly, maybe it helps to dislodge something sticky like peanut butter that gets attached to the lumen. So it can help to dislodge food. It can change the surface area of it to increase and decrease absorption. Absolutely. So it can play some role that way. Excellent. So we have this first layer, our mucosa, that is comprised of these three distinct layers. Now, deep to the mucosa, not surprisingly, is the submucosa. The submucosa is also comprised of areolar connective tissue. Does that make this a lamina propria? No, exactly. Right. Remember, a lamina propria is an areolar connective tissue that is part of a mucous membrane. This is an areolar connective tissue, but it's not a part of the mucous membrane. Instead, this is the location where we're going to find, large glands and blood vessels. Oops, what's going on here? Oops, sorry. So we'll find large glands and blood vessels in this region, whereas our lamina propria is going to have small glands. It's going to have capillaries. It's going to have malts, right? Our mucosa-associated lymphoid tissues. In fact, if we cheat and go back to our pretty picture, notice here in green, they've actually given us a couple of lymphatic follicles here in our lamina propria, helping to provide that protection from things from the outside world. Excellent. Questions on that? All right, perfect. Our third layer, and I need to leave a little bit of room here. I'll talk about why in just a second, is what is known as the muscularis. Now notice the muscularis mucosa was the name of the thin layer of smooth muscle in our mucosa. So calling this region the muscularis is perfectly acceptable, but sometimes it is also referred to as the muscularis externa. Our muscularis externa is actually comprised of two layers of smooth muscle. And these two layers are perpendicular to each other. So layer 1 and layer 2. Oops, hold on. I need to cheat again. Bring that down. Bring that down. Perfect. The innermost layer, the layer that is closest to the lumen, is a circular layer of smooth muscle. basically the circular layer goes around in a circle around the circumference of our organ, the lumen of the organ. And so if you think about it, if we were to change or contract this circular layer, what effect is it going to have?
Making the lumen smaller.
Yeah, or if it contracts, absolutely, and if it relaxes, it's going to dilate it. So its effect is going to be to change the diameter of the organ. And the outer layer is a longitudinal layer. And so what is its effect going to be? When this layer, this longitudinal layer that runs the length of the organ, when it contracts, what's going to happen?
Shortens the whole length.
Yeah, it's going to shorten the organ. When it relaxes, it's going to lengthen the organ. So its effect is going to be to change the length of the organ. All right. Now notice if we start putting these together in a rhythmic fashion, contract the diameter, then shorten, contract the diameter, then shorten, contract the diameter, then shorten. Notice this type of activity in a rhythmic fashion, these rhythmic propulsion of these two layers could lead to the movement of substances from one location to the other, which, as Izzy pointed out, we call peristalsis. And that's indeed what peristalsis is, an alternating contraction of these circular and longitudinal layers to propulse the food from one location to another. All right. Questions on that? All right, excellent. Now, notice, remember, we said the basic functions of the alimentary canal are to have glandular activity, change the motility of our muscles. And as we said, to do that, we need to use hormones and we need to use nerves. Remember, we have that big, elaborate network of nerves that is called the enteric plexus. Again, that enteric plexus is an elaborate network of nerves and ganglia that are going to give us local control of our alimentary canal. And as it turns out, there are two main branches of the enteric plexus. One of those main branches happens to be located in the submucosa and is known as the mucosal plexus, submucosal plexus. So here in our submucosa, there's a submucosal plexus, and guess what it controls? If it's in the submucosa.
Nerves in the submucosa.
Yeah, the glands of the submucosa. So here inside of our submucosal layer, we're going to have some nerves that are going to control those glands. And we're also going to have some nerves that control the layers of the smooth muscle. And so these are known as the myenteric. And the second branch of our enteric plexus is the myenteric plexus, and it controls the smooth muscle. Again, I appreciate that my picture is not the prettiest, but if we cheat and come back to this picture, notice, lo and behold, when we look at this pretty picture here, in the mucosa, we see all of these yellow nerves that are located in here. And notice up here, and that is the submucosal nerve plexus. And notice up here in the muscularis externa within the smooth muscle, we have our myenteric plexus. So notice that enteric nerve plexus That intrinsic nerve plexus is made up of the myenteric and the submucosal. Submucosal controls the large glands and blood vessels. The myenteric controls the muscle. All right. Questions on that? All right. That brings us to our fourth layer. Oops, hold on. I want this to be black. I want this to be 24. Our fourth layer. Notice, as we know, let's do it this way. Most organs of the alimentary canal are contained within the abdominopelvic cavity. That is an important piece of information that tells us a couple of things. One of the things that it tells us is that since the abdominopelvic cavity is open to the outside world, what should the outer surface of our alimentary canal be lined with? What type of membrane? Not a mucous membrane, but what type of membranes line surfaces that are closed to the outside world?
Cirrus?
Yeah, exactly. So it should be by a serous membrane, or again, serous is the adjective that describes the noun membrane. We can say serosa. And we can even be more specific than that, because we know if this is a serous membrane that is lining the surface of organs in the abdominal pelvic cavity, how would we describe that specific serous membrane?
Visceral, serous
Visceral what?
membrane.
True, but there's a name for the membrane.
Peritoneum.
Peritoneum, excellent. Excellent. So that serous membrane could also, or serosa, let's actually do it this way, could also be known as the visceral peritoneum. Excellent. And that's a good thing, because as a visceral peritoneum, as a serous membrane, we know, like all membranes, it's comprised of an epithelial tissue sitting on top of a connective tissue. And what epithelial tissue would that be?
Simple squamous.
Simple squamous. Epithelial tissue. And what connective tissue would it be?
Dense irregular.
Not dense or regular. What connective tissue sits under the simple squamous of a serosa? Areolar, excellent. So in this case, we would have, and let's use, we haven't used green yet, a single layer of flat cells sitting on top of an areolar connective tissue. There, give me nuclei. Excellent. Questions on that? No questions? Because I have one. Actually, I have two. First, why didn't I draw that all the way across? Did I just get lazy? And secondly, what it says most, and what does most mean?
Not all.
All.
Not all. Notice most organs of the alimentary canal are contained within the abdominal pelvic cavity, but some organs of the alimentary canal are not. Oops. Some organs are not contained inside of the abdominal pelvic cavity. And those that are not contained inside of the abdominal pelvic cavity don't have a serous membrane. So instead, their fourth layer, their outer layer, is what is known as an adventitia. And that adventitia is basically just a dense irregular connective tissue that basically attaches it to the surrounding tissue. Right? Again, if you think about it, one of the advantages of that serous membrane, its function is to reduce friction and allows movement, right? This one allows movement. However, if we've got instead an adventitia, we've just got this very fibrous connective tissue that is anchoring it and holding it in place. And when it anchors it and holds it in place, it's obviously not free to move around. And so a organ will either have an adventitia or a serous membrane. It won't have both in any one location. Let's take a look at this now with the pretty pictures. Hold on, before we do this, I'll save this picture so I don't forget. So again, our innermost layer, closest to the lumen, is a mucosa, has 3 specific sublayers. Our simple columnar epithelial tissue, our lamina propria, which is an areolar connective tissue containing small glands, capillaries, and malts, and a thin layer of smooth muscle known as the muscularis mucosa. And again, this can help change the shape of the lumen, the surface area of the lumen, dislodge things from the surface of the lumen as it moves that way. And we did that. Deep to that, we have our submucosa. There it is. Submucosa is also areolar connective tissue. But again, it's not a lamina propria because it's not a part of the mucous membrane. Notice this is areolar connective tissue. This is areolar connective tissue. There's really no difference between these tissues. The only difference is the location. One is inside the muscularis mucosa. One is outside the muscularis mucosa. So what's outside the muscularis mucosa is the submucosa, containing large blood vessels, large glands, and that submucosal nerve plexus. Our muscularis, or muscularis externa, again, both are acceptable names for this, is comprised of 2 layers. And I think it's important to take a note of this. Let's take a quick look at this right here. Notice the 2 layers are perpendicular to each other. But how they appear under the microscope can vary depending on the plane of section. Notice when we do a cross section through the gland, we see the circular layer as being longitudinal. However, notice when we take a lengthwise cut through the organ, we see the circular layer as being, actually, let's change that, as being a cross section. Notice with a transverse section, the longitudinal layer is in cross section. However, in a lengthwise section, the longitudinal layer is a longitudinal view. So notice you can't tell which is circular and which is longitudinal by their plane of section, whether they're cut on edge or whether they're cut lengthwise. You can tell them by their location. The one that's closest to the lumen is the circular. The one that's further from the lumen is the longitudinal. And no matter what, they're always going to be perpendicular. So it's going to always be easy to tell the two layers from each other because they're perpendicular. So one will be in cross section, one will be a longitudinal section. So you can't tell by the plane of section, but you can tell whichever one's closest to the lumen is circular, and whichever one is closest to the outer surface is the longitudinal. Now, remember, as we mentioned, we are going to, this is controlled by our myenteric nerve plexus. And this is responsible for that peristalsis. Here's a really basic picture of this peristalsis. I like this one from your textbook a little bit better. Notice again, rhythmic alternating contractions of circular layer, then longitudinal layer, then circular layer, then longitudinal layer. And in that, we get that forward movement of our food from the mouth towards the anus. And this rhythmic alternating contractions of the smooth muscle is what we call peristalsis. However, peristalsis is not the only thing this smooth muscle can do. Let's look at another type of movement that can be caused by our smooth muscle. This particular motion is called segmentation. Notice what's happening here is we're getting an alternating contraction of the circular layers. First, this part of the circular layer contracts, then that one, then back and forth and back and forth and back and forth. Notice with this type of alternating contractions, are we getting a lot of movement from left to right or from top to bottom in this here? No. So notice this one isn't about propulsion. Instead, when you're mashing this stuff back and forth, this segmentation is really about two other processes. One is our mechanical digestion, because as we pointed out, while everybody in class here chews their food the 25 times before they swallow, some people take a big, huge, massive bite, chew something twice, and then swallow it down into their gullet. So in which case, things like our stomach, things like our small intestine need to increase the mechanical breakdown of that food. And so this type of alternating contraction does that. It also helps to mix it with the enzymes for the chemical breakdown. And in the small intestine, it also rubs the food back and forth across the surface of the small intestine, which increases the absorption. So notice this segmentation is a very important function, but it's not about propulsion. And there are lots of other examples about this as well. When we get to the large intestine, we'll talk about haustral contractions. We'll talk about mass movements, right? We'll talk about movements in the stomach, the churning, right? There's all sorts of different types of smooth muscle contractions. Peristalsis is a big one, but it's not the only one. Lastly, we have our serosa, and notice this one indeed is a serosa. We can see a simple squamous epithelial tissue on top of an areolar connective tissue. But this doesn't show it, but sometimes we also have an adventatia where it'd be anchored in place. All right, we need to talk a little bit more about this adventatia, and we also need to talk a little bit about mesenteries. But before we do that, any questions on the basic anatomy of our alimentary canal?
I just want to double check the smooth muscle contractions you were talking about were part of the muscularis layer movement.
Yes. So all of the muscularis layers are what's responsible for peristalsis and most of the other smooth muscle contractions. Remember that as we talked about, the muscularis mucosa really is about just the surface area of the small intestine. I mean, of the lumen and dislodging of any materials. All right, excellent. Tell you what, there's a couple, like I said, I want to talk about mesenteries. I want to talk adventatia. There's a couple more things that I want to talk about, but we've gone a little long, so I think this is a good stopping point. No, mesentery is not adventatia. We'll talk about what the mesentery is. We'll talk about what adventatia is. We'll talk about those in a second, but I think now is a good time for our first break, because like I said, we've gone a little bit longer than I would have liked to. But let's come back and do this after the break. So let's go ahead and take our break, and then we'll come back and talk about the mesenteries, talk more about the adventatia, when you use when, and at which, and then we'll do that. But let's do that after the break. So we will restart at 9:35, and I will start the recording at that point. So we will come back and pick up from here. Any questions? All right, see you in 15 minutes. So to continue with where we're going, I want to do a very quick review of what we've been talking about and kind of look at it from a different point of view. Right here, as you see during the break, I spent almost the entire break drawing this amazing recreation of what the abdominal pelvic cavity looks like. And our job here, notice there's a big empty space in the middle, and we're just basically going to draw a stomach, right? Obviously, this is not going to be to scale, but we get the point. And of course, the stomach starts with the innermost layer, which, as we know, is that mucous membrane. So we know that mucus, nope, hold on, I want this to be purple. This mucous membrane starts at the outside, and then it comes to our stomach. And as a stomach, as it turns out, has a big convex portion that comes out, and again has a concave portion that comes out. And then it connects to the small intestine, which of course then, oops, there you go, you get the idea, leads to that there. So obviously the first part is indeed that mucous membrane, or the mucosa. Actually, mucosa fits better, so let's do that. And we know that mucosa basically is contained of three layers: a simple columnar, epithelial tissue, and areolar connective tissue, and our muscularis mucosa. So we can cheat a little bit on this. I'm not going to take the time to draw all of it, but we can draw enough of it that we have a simple columnar epithelial tissue, sitting on top of that areolar connective tissue that we know then sits on top of a layer of smooth muscle that is the muscularis mucosa. Oops. And we'll pretend that was on the outside like it belongs. Excellent. We then know the next layer is our submucosa. And that submucosa is just areolar connective tissue. And so we have to draw that, and I'll cheat and just do this as a highlighter. And again, this areolar connective tissue is no different from the areolar connective tissue that's inside of the mucosa. They're both areolar connective tissues. It is just not a part of the mucous membrane, so it's not a lamina propria. Of course, we then have our muscularis externa. Again, we can use the term externa or not. Both are acceptable. And it is two layers of smooth muscle. And we can draw those. Layer one, layer two. And of course, we know that the one closest to the lumen is the circular, which is closest to lumen. And then our longitudinal, which is furthest from the lumen. And that these two layers are perpendicular to each other. And then for most organs like our stomach here, as we mentioned, we are going to have a serosa, which, as we know, is the visceral peritoneum, which we know is a simple squamous epithelial tissue sitting on top of an areolar connective tissue. So again, we have this thin layer of areolar connective tissue, no different from any of the other areolar connective tissues that we've had, with the exception of its location. And then, of course, a single flat layer of squamous cells. Now, we know one other thing about this visceral peritoneum. We know that it is continuous with the peritoneum that lines the entire abdominal pelvic cavity with an areolar connective tissue and a simple squamous epithelial tissue. So notice, not only have we now made a stomach, from inside to out, we have made an entire stomach, but we've also reminded ourselves that while this one here, because of its location, we call the visceral peritoneum, we also know that it is continuous with this layer that lines the cavity of the abdominal pelvic cavity. And what would we call this layer of serosa that lines the cavity of the abdominal pelvic cavity?
Parietal.
Parietal.
Right. Excellent. So notice we've put our layers together. We have made our stomach from inside to out. And we've reminded ourselves that our visceral peritoneum, that serosa on the outer surface of our stomach, is continuous with the layer that lines the outer cavity of the abdominal pelvic cavity, the parietal peritoneum. And that works fine with this simple illustration, but we have one more issue. Because what if instead we took a cross section? And in that cross section, like we had here, we had our small intestine in this case, in our abdominal pelvic cavity. Whoops. Of course, we know that our small intestine needs to be lined with an areolar connective tissue and needs to be lined with a simple squamous epithelial tissue that we know is its serosa on the outer surface. So again, we have our simple squamous, but we also know that this needs to be continuous with the parietal peritoneum. Lining the cavity of our abdominal pelvic cavity, we have an areolar connective tissue. We have a simple squamous epithelial tissue. That again, collectively, we call the parietal. Oops. Spelled correctly. Which also is a simple squamous. Epithelial tissue and areolar connective tissue. And so we know these two things need to be continuous. So as it turns out, what happens is that this simple squamous epithelial tissue will actually come off of the posterior wall of the abdominal pelvic cavity to extend up to the organ like the small intestine, wrap around the organ of the small intestine, and then it comes back down to connect to the simple squamous on the other side. Of course, that means the inside of this is going to be lined with areolar connective tissue as well. And notice basically what we have here back to back is a back to back double membrane of serous, a double serous membrane. Let's say it that way. We actually give a fancy name to this back-to-back double serous membrane. Anybody know what we call this back-to-back double serous membrane?
Mesentery.
Exactly. This is the mesentery that someone was asking, Izzy was asking about before the break. A mesentery is a back-to-back double membrane of serous membrane. Notice it's a simple squamous epithelial tissue with a double stuff Oreo with 2 layers of areolar connective tissue mashed together in between. This loosely connects the intestine to the posterior wall of the abdominal pelvic cavity, minorly restricting it, but allowing some freedom of movement. But the other huge advantage of this is going to be a pathway also for blood vessels, lymphatic vessels, and nerves. So it's a pathway for blood vessels, lymphatic vessels, and nerves. And this is what our mesentery is. So notice if we go back to the pretty picture from our textbook, we can actually see how that serous membrane that wraps around it from one side and wraps around it from the other side comes together to form that double membrane formed by 2 simple squamous epithelial tissues and an areolar connective tissue in between, and forms a passageway for blood vessels, lymphatic vessels, and nerves. And that structure is the mesentery. So mesentery is a double sheet of the peritoneal membrane. Mesentery is a generic term. So there are many mesenteries. However, are you going to need to know specific names for specific mesenteries? Absolutely. So these mesenteries are access routes for blood vessels, nerves, and lymphatic vessels. And one of the ways we can see this is it allows us to be able to, we'll see them and identify them in some of the views. But as I mentioned, not only is it a passageway for blood vessels and nerves, but it also helps to stabilize the organs while still allowing some movement. Back in ancient times, there were these things called earbuds, right? Earbuds were things that would allow you to listen to music through them by connecting them to things like a Walkman or even things like your phone, right? I know now we all have the wireless ones, but they used to have wires connected to them. And there was something very magical about these earbuds. You could take these earbuds and their cords, put them into your pocket, and immediately take them out. And when you did that, what happened to the cords in that 1 second they were in your pocket?
Tangled.
Yeah, they all became completely entangled. Is that what you want happening to your small intestine every time you eat a cheeseburger? Do you want it to tie itself up into knots? No. So it's going to help to limit the mobility of the organs without stopping it entirely. So not only is it an important passageway, but it's going to provide some structure, right? Let's take a look at the pretty picture here to see this. Notice most of the organs of the abdominal pelvic cavity, like the liver, our stomach, our large intestine, our small intestine, we can see these membranes. Notice they've done a really nice job of this. Notice in green, they have shown us our parietal peritoneum lining the outer cavity of the abdominal pelvic cavity, right? Notice in light blue, let's make this a little smaller. They have shown us the visceral peritoneum on the surface of the organs. But notice connecting them, they have shown us different examples of our mesenteries. Notice there is a mesentery called the lesser omentum that connects the liver to the to the stomach. Anytime there's a lesser something, there is going to be a greater something. And the greater omentum comes off the front of the stomach, forming this big, long, drape-like apron over the front of the abdominal pelvic cavity, and then connects to our large intestine. Notice our small intestine are held in place by mesenteries. Our large intestine, our transverse colon, is held in place. And we can start to see some of the names of some of these mesenteries. So again, mesentery is a generic term, and here we can see some specific examples. The lesser omentum, as we talked about, connects the liver to the stomach. The greater omentum connects the stomach to the transverse colon, but notice also forms this long drape-like structure in front of it. The transverse colon of the large intestine is anchored in place by the transverse mesocolon. Notice the small intestines are held in place by structures they just have labeled as mesentery. Now remember, we just learned mesentery is a generic term. So to distinguish these specific mesenteries that hold the small intestine in place, we will refer to them as the mesentery proper. So don't just say mesentery if I ask you to identify these, they are the mesentery proper, and we can see that there. Let's change the view a little bit. Notice here, they've taken that greater omentum, that drape, and lifted it up where we can see where it connects to the large intestine, specifically the transverse colon of the large intestine. This also allows us to see where the transverse colon is connected to the back wall, the transverse mesocolon. Notice also this S-shaped portion of the large intestine called the sigmoid colon is also anchored by a mesentery that we call the sigmoid mesencolon. And again, here holding the small intestine in place are the mesentery proper. So those are all the mesenteries you're responsible for. Your textbook does a nice job of showing these. When you come to the Cumnus River College site and look at the digestive charts, when we get to the actual digestive one, come on, there we go. Notice we can see a little bit of the greater omentum draping down here. Notice they've cut the transverse mesocolon that holds the transverse colon in place. So we see that really nicely. This isn't the picture that I wanted though. Clear these drawings. Now this shows it a little better. You can see the greater omentum draping down, and here's the mesentery proper, transverse mesocolon. But that's still not the picture I wanted. They have the picture I wanted? This helps. Actually, this is the one I wanted. Perfect. Notice this one here. And again, I know we haven't talked about the anatomy of the stomach yet, but when we get to the anatomy of the stomach, notice there, as we talked about, is a concave curve and a convex curve to the stomach. The concave curve is known as the lesser curvature. And notice the lesser curvature is where you find the lesser omentum. The greater curvature, and even though it's cut on this illustration, is where the greater omentum comes down. So the greater omentum and lesser omentum come off the lesser curvature and the greater curvature of the stomach. That makes it convenient. Again, we see that cut transverse mesocolon that anchors and holds the transverse colon in place. Here we see the sigmoid mesocolon that holds the S-shaped sigmoid colon. And notice we can see the cut mesentery proper that holds the small intestines in place. So notice we see lots of examples of these specific mesenteries that can be seen in lots of locations. All right, questions on your mesenteries? Oh, here's another pretty picture from your textbook. Does a great job of showing that lesser omentum connecting the liver to the lesser curvature of the stomach. And again, a cut, greater omentum. Questions on that? Because if this doesn't make sense, it's just going to get worse. All right, either my speaker's not working again or nobody is speaking. All right, excellent. So in that case, then let's go back a couple pictures to this one. Because notice there's one more thing that I want you to notice about this illustration. Notice, for instance, there are a couple organs. One of those organs, for instance, happens to be the pancreas, is located actually behind the abdominal pelvic cavity. It's not actually located in the abdominal pelvic cavity. It's behind the parietal peritoneum. What actually happens, and let's cheat and go back to our illustration. I think I've saved this picture. Yep, I have saved it. So let's cheat and do this. What actually happens is that there are some organs. Actually, I need more space. There are some organs that are actually going to be embedded in the wall of the abdominal pelvic cavity and actually will be behind the parietal peritoneum. So notice there's my parietal peritoneum. Here is my simple squamous epithelial tissue, that's serosa. And organs that are located behind the parietal peritoneum are given a special name. They're not in the, or like this one here that is in the abdominal pelvic cavity, we call intraperitoneal. Whereas ones that are outside the abdominal pelvic cavity, oops, peritoneal, what would we call those? Come on, I know you know, even if you don't know, you know. What do we call an organ that's not in the abdominal pelvic cavity, behind it? There you go, retroperitoneal. Give me some examples of some organs that you know or think might be retroperitoneal. Excellent. Kidney is definitely one of them. And if the kidney is definitely one of them, someone remind me again what sits right on top of the kidney? Adrenal gland, absolutely. However, the adrenal gland, because it sits on top of the kidney, is sometimes referred to as the suprarenal gland. But you are right, that is also the adrenal gland. Give me another one. Pancreas. Pancreas is actually interesting. The pancreas is a feather-shaped organ. It has a head, it has a body, and a tail. For the pancreas, actually, the head and the body are retroperitoneal, where the tail actually sticks out into the abdominal pelvic cavity. So most of the pancreas is retroperitoneal, but not all of it. And that brings us to a couple other good points. As someone just mentioned out, not all of the small intestine is located inside the abdominal pelvic cavity. In fact, the proximal beginning part of the small intestine, a portion known as the duodenum or duodenum, both are acceptable terms. The duodenum of the small intestine is located in the abdominal pelvic outside of the abdominal pelvic cavity. So the duodenum is retroperitoneal. Notice for the large intestine, the transverse colon has a mesentery, the sigmoid colon has a mesentery, but the part of the colon that comes up and the part of the colon that comes down don't. So those are the ascending and descending colon. So for our colon, the ascending and descending colons are retroperitoneal. What else can you think about that as you look at the body would be located in the back part of the body, anchored and wobbling in place? What about large blood vessels? You learned about some really big, large blood vessels. What are the largest blood vessels of the abdominal pelvic cavity? Do you want those wobbling around? If only I had learned the blood vessels at some point in this class. The aorta, and what's the other one?
Superior, inferior vena cava.
Excellent. They happen to sit right next to a muscular tube that travels from the thoracic cavity to the abdominal pelvic cavity. What might that be?
Esophagus.
Esophagus. Let's go back to the kidneys. The kidneys are anchored in the back wall. They need to be able to communicate to the bladder, carry urine to the bladder. What were the organs that allow the kidney to carry the urine to the bladder? Anybody know?
Ureter.
Ureters. Important for the removal of waste. And also the distal part of the digestive system is also important for the removal of waste, the rectum. These organs are your retroperitoneal organs. And notice if they're retroperitoneal, they are not going to have a serous membrane that lines their surface. Instead, they're going to have a fibrous connective tissue that anchors them to the posterior wall. So these retroperitoneal organs are the organs that have adventitia. So organs that are retroperitoneal have adventitia. Organs that are intraperitoneal have serosa. So that's why you can have one or you can have the other, but you can't have both. Because you're either retroperitoneal or you're intraperitoneal. So you either have an adventitia or you have a serosa. And remember, if you have trouble remembering which organs are retroperitoneal, just remember that if you're retroperitoneal, you are a sad pucker. PP, pucker. I said pucker. Pucker. All right. Questions on that? Oh, no, wait, wrong button. Sorry. Meant to save. All right. So again, I have a pretty picture here that shows this as well. Notice here we see intraperitoneal organs located inside the abdominal pelvic cavity. They typically have mesenteries. whereas others may even start in the abdominal pelvic cavity, but are brought back into a retroperitoneal location behind the parietal peritoneum. All righty. Questions on that? All right, excellent. With this, then, we have our basic anatomy, basic control. So now we can start getting into specifics. As I mentioned, unlike previous sections, we are going to work our way down that mystical, magical journey that is the alimentary canal, starting first in the oral cavity, the mouth. Oral or buccal cavity. What forms the oral or buccal cavity? Let's think about this. What are the boundaries? We can make this easier if you want. You can just rattle things off, or we can kind of think of it in terms of where its locations are. So what are some of the things that form the boundaries of our oral cavity? Excellent. So anteriorly, we have the labia, superior and inferior labia, or the lips that we think. Excellent, right? So we have those. We have the cheeks laterally. However, we have no more about that. Obviously, there's skin located there, but what is the primary muscle that forms the cheek region of the oral cavity?
Buckle?
Close. What was it called again?
Buccinator.
Buccinator. Excellent. Buccinator or buccinator. Absolutely. The buccinators. Absolutely. Right. And as well as a little bit of the temporalis, masseter, stuff like that. So we have some of the lateral muscles as well, but I think the buccinator is primarily the primary one. Excellent. What would you consider to be the superior boundary of the oral cavity?
Palate.
All right, excellent. So the skull feature, the hard palate, which indeed is a skull feature. Of course, like all skull features, you know it is comprised of bones and bone features. So someone remind me what the 4 bones and bone features that form the hard palate are. All right, excellent. So the right palatine bone, the left palatine bone. What else? What other bones? I always think it's adorable when you guys think that the stuff from 430 isn't relevant for 431. There you go, the maxilla. How many maxillary bones do you have again? I heard 2, I heard 1, which is correct?
1.
1?
2 left and right.
There you go. We have indeed a right maxilla bone and a left maxilla bone. Of course, way back in 430, we had to learn the bone features that actually made these as well. So that would probably be useful. What portion of the palatine bone actually helps to form the hard palate? Remember, the palatine bone was kind of this C-shape or L-shaped bone, and there was this bone feature that ran parallel to the ground, kind of in a horizontal orientation. Anybody remember what that bone feature was called? Come on, peeps, I just gave it to you. What was that horizontal bone feature called?
Skull.
How about the horizontal plate? Can anyone remember the bone feature of the maxillary bones that connected to the palatine bone? The stuff's going to be on the exam, so if you didn't learn it in 430, it's time to learn it now. Palatine process. So the roof of your mouth bone features, right? We have the skull feature, the hard palate is made up of the right palatine bones horizontal plate, the left palatine bones horizontal plate, the right maxillary bones palatine process, and the left maxillary bones palatine process. Right? You should remember and know that from looking at the bones. If you don't, here, let me cheat. There we go. This looking vaguely familiar? Again, this whole collective structure is known as the hard palate, formed by the sutures between 4 different bones and bone features. Right? That is the palatine process of the right maxilla, palatine process of the left maxilla, horizontal process of the right palatine bone, right? Horizontal process of the left palatine bone. Back in 430, we talked about in some individuals, especially if their moms didn't have enough folic acid, you will not get a closing of this suture. What do we call that condition?
Cleft lip.
Yeah, cleft palate in this case, absolutely a cleft palate that we see from that there, which luckily can be fixed surgically. Is this the only thing that forms the dividing structure between the oral and nasal cavity? Oh man, we went away from here, so I've lost all of it. Superior and inferior labia. What muscles form the superior and inferior labia primarily?
Orbicularis oris.
There you go. Lateral, we said the buccinator, plus other muscles. We were working on superior, which I won't bother writing out again, but we know is the hard palate. But definitely you are responsible for the individual bones and bone features that form the hard palate. Is that the only thing that helps to form the superior boundary between the oral and nasal cavity? Well, I'm asking the question, what's the obvious answer? No. So what else helps to form the boundary? Come on, you guys were all singing about it this summer. This fall, winter, whatever month, I don't know. I don't have any idea what season it is. It's that little thing dangling in the back of your throat.
Tongue.
Yeah, the soft palate. We have the soft palate, connective tissues, and other structures that terminate at the uvula. Right, the dangly thing that swings in the back of your throat. Excellent. We did that. Anterior, lateral, superior, inferior. I think I saw someone write tongue way back in the day. I accept that to some extent. Anything else we could throw in there as well? What else helps to form the lower part of your oral cavity? What else helps to form? Well, remember the tongue has intrinsic and extrinsic muscles. But what else forms a boundary at the lower part of your mouth? Mandible. There you go. See how easy that was? I didn't even have to give you any hints. I heard myself doing it, but all right, excellent. There you go. So we have this oral cavity, absolutely, with all of the fun anatomy that goes along with it, right? As we mentioned, it has to deal with that hot coffee, that acidic wine, those spicy tortilla chips, those sharp tortilla chips, a tremendous amount of use and abuse. So what type of epithelial tissue forms the mucous membrane of our oral cavity? There we go. Non-keratinized, stratified squamous. Epithelial tissue, excellent, so that it can take all of that use and abuse. Absolutely. However, and again, this is one of those things where it's easier when we're all in the classroom because I tell you to grab the person who's sitting next to you in class and stare inside of their mouth. Can't do that now, but if you happen to have other living individuals in the house with you at this point of time, grab them at some random point during the day, look inside their mouth. And when you do that, you may notice along the gums or sometimes on the roof of the mouth or on the surface of the tongue in some areas, you might see a slight whitish coloration. In that whitish coloration occurs because of the increased friction in the oral cavity. Increased friction or increased damage from spicy food or sharp foods or hot foods, things along those lines, can actually cause a minor keratinization of the tissues. So that little bit of whitishness that you see on the tongues or on the gums can be a slight amount of keratin that is forming from friction or abuse or temperature or things along those lines. Here we have the pretty picture showing all of the things that we've talked about. Notice we have the superior and inferior labia, the lips, and again, the lip is that whole flappy structure, right? Normally when we think of the lips, we think of just where you're putting your lipstick. However, where you put your lipstick is really a portion of the labia that we know as the red margin. The red margin is this transition area between the keratinized stratified squamous of our skin and the non-keratinized stratified squamous of our inner surface of our oral cavity. In this region, the stratified squamous epithelial tissue becomes very thin. It makes the region more sensitive, especially to things like temperature and abrasion. It also, because it's thinner, we can more clearly see the blood vessels underneath, which is what gives it its reddish coloration, why it's called the red margin. It also makes it a good indicator point if someone is lacking oxygen or too cold or jaundice or something along those lines. That can be seen more clearly in those regions. Not only is the epithelial tissue thinner in that red margin, but typically there are no hairs and no glands in that area. Obviously, you knew about the no hair because you've never had to shave your actual lips, the red margin. But how did you know that it doesn't have glands on it? How do we moisten and maintain that tissue if it doesn't have glands?
Chapstick.
True, okay, but what did cavemen do before chapstick was invented? Right?
Lick
Burt's
your
Bees
lips.
has been around a long time, but it hasn't been around forever. What did we do before that? There you go. Licked our lips, absolutely, right? We licked our lips to do that. Excellent. And of course, the primary muscle of the labia is the orbicularis oris, the buccinators. For that, we did all of that. Notice the other thing that you could kind of argue is the anterior and lateral boundaries of the oral cavity would be the teeth. Remember, the teeth are bone-like structures, but they're not actual bones. However, notice there is a small space in between the labia and the teeth, basically this indentation where the gums and stuff like that is. This region here is known as the vestibule. Again, if you think back to middle school when you first started chewing tobacco, that would be where you take your big chaw and stick it right into that vestibule, into that space. Causes someone to eat on their lips or the inside of the mouth. I wouldn't say it's a disease. I think so I'd say two things. It is not uncommon for people to accidentally bite their lip or bite their inside of their cheek or something like that while they're eating. However, it could also be a nervous activity, the same thing as biting your fingernails or toenails or whatever you chew on. So, I mean, again, there can be nervous habits that cause things like that, but I wouldn't say that it is a disease. Now, notice also, we don't want, again, this whole upper part, this whole lower part are the labia, and we don't want them flapping around too much. So notice you will see that there is a fold in the mucous membrane that anchors the labia to the vestibule. So superiorly, that is the superior labial frenulum and the inferior labial frenulum. So those frenulums help to stabilize the labia and hold them in place. Again, notice here in the oral cavity, we see the four bones forming the hard palate. We see the connective tissues forming the soft palate, tapering down to the uvula. And you could argue the tongue or the mandible is the inferior boundary of the oral cavity. Notice we've talked about this once more because if you remember, we were talking about one other key feature here in the oral cavity. Remember, we talked about these two arches. One of these arches goes from the palate, the roof of the mouth, towards the tongue, known as the palatoglossal arch. The other arch goes from the hard palate, or the soft palate, I should say, down towards the pharynx, the throat. And so that is the palatopharyngeal arch. And remember, we mentioned how the space in between is what is known as the fosse. And inside that fosse, remember, is where we found our palatine tonsils. So we see this nice anatomy as we look into the oral cavity. Notice here, we see this as well. Someone was talking about the intrinsic and extrinsic muscles of the tongue that change the shape of the tongue and move the tongue around in space. So you could see it kind of forms the inferior boundary of the oral cavity. But you could also argue that it's in the oral cavity, so I wouldn't have a problem with you saying the mandible was the floor. Here we see the hard palate and the soft palate, the vestibules, and the teeth. We see all the boundaries, and notice there really isn't a posterior boundary. It basically feeds straight into the pharynx. All right, questions on the basic anatomy of the oral cavity? Excellent. So we talked about basically 6, 6.5, 7, however you want to think of, functions to the digestive system. How many of those take place in the oral cavity? Any of them?
The entrance,
Okay, obviously ingestion. And unless you have a feeding tube, this is the only organ where ingestion takes place, right? So this is where, again, food first enters in mostly a conscious effort. Although sometimes when you're sitting there watching a movie, you can be unconsciously shoveling popcorn in there. Well, let's see if that's correct, Izzy. I think you got the right idea. Obviously, defecation does not occur in the oral cavity, but let's talk about the other ones. So you said, but that's not defecation. That may be the emission of materials, but it is not, that wouldn't be defecation. That would be a reverse emesis or ejection of food. So we talked about mechanical digestion. Does that occur in the oral cavity?
chewing.
Yeah, absolutely. Chewing or, again, mastication. Absolutely. The physical breaking down of food stuff. Absolutely. What were the other functions?
Chemical.
Chemical. Chemical digestion, of course, involves enzymes. Are there enzymes that are produced in the oral cavity?
In the saliva.
Yeah, absolutely, right? Remember, we also talked about secretion taking place, and absolutely we secrete saliva. We'll talk about the function of saliva in a little bit, but chemical digestion, one enzyme that is produced in the saliva is salivary amylase. Anyone know what salivary amylase helps us break down?
Dark foods.
Yeah, carbohydrates. You can actually experience this for yourself. During the next break, go take a paper towel. Where are we on time? Yeah, we're going to go down to your kitchen, take a paper towel, dry off the surface of your tongue, take a little square of white bread and stick it on the surface of your tongue and just let it sit there. As your tongue and your mouth produce more saliva, that saliva is going to mix with that bread, start to break down the carbohydrates into simple sugars, and you will actually start to taste sweetness on the surface of your tongue from that breakdown of the carbohydrates. Now, it turns out this isn't the only enzyme that is produced by the oral cavity and in the saliva. We also have, from our tongue, a lingual lipase. Guess what lingual lipase helps us to break down?
Lipids.
Lipids. So does that mean if I took a little chunk of butter and I stuck it on my tongue and allowed the saliva to mix with it, I would start to taste the acidity of the fatty acids or the sharpness of the alcohol from the breakdown of those fats?
It's
No.
butter. You would taste heaven.
And turns out, no, it doesn't. In fact, because it is actually released in an inactive form. This lingual lipase doesn't actually become active until it reaches your stomach. Why is it active in your stomach but not active in your mouth? What's the big difference between your stomach and your mouth? There you go, pH. What's the pH in your oral cavity? Yes.
High, basic.
Well, it's actually pretty neutral. It's around 7. What's the pH in your stomach?
Low.
Very low, about 1 or 2. Absolutely. So the change in pH causes the enzyme to form a conformational change, and it actually becomes active in the stomach. So again, it won't break down that lipid on your tongue, but it is released in an active form. So it is released in there as well. So definitely some chemical digestion takes place. Propulsion. Does propulsion take place in the oral cavity?
Your tongue starts pushing the food back.
Yeah, swallowing, or the process we call deglutition. That swallowing absolutely is an act of propulsion, moving it from the mouth into the next organ in the alimentary canal. And what does that leave us with? It's 4. We know defecation doesn't occur there. So what's the last one?
Absorption.
Does absorption occur in the oral cavity? Yeah, it's limited, but it does, right? Again, you may not have thought of it in these terms, but when grandpa comes to visit right after he gets his COVID shot and you get your COVID shot, eventually grandpa's going to be able to come and visit again. And grandpa has that heart problem. And every once in a while, when his heart starts to fluttering, you got to give him medicine. What kind of medicine do you have to give him?
Nitro.
And where do you put that nitro pill?
Sublingual.
Yeah, under the tongue. Absolutely. So some medicines, some alcohols, some water, some other basic components. There is some limited absorption that can take place in the oral cavity. Absolutely. Obviously, it's not the primary location for absorption, but there is some limited absorption that can take place in the oral cavity. So Izzy had it 100% correct, right? We can do all the functions except for defecation. And don't think I've forgotten your comment about lobsters, Timothy. Is that really what lobsters do? Do lobsters only have a one opening to their digestive system?
Oh, that's just something I heard. I don't know if that's true.
If it is, that's incredibly gross. I may stop eating lobsters if that's the case. I mean, not that I do it frequently anyway, but you get the idea. So absolutely, yes. We have ingestion, mechanical digestion, secretion, chemical digestion, absorption, basically and propulsion. And that doesn't even include our other major non-digestive function. But obviously it helps to assist in digestion. And so maybe instead of non-digestive, we can say accessory digestive function. And that is what? What primary sensory input do we get from the oral cavity that assists in digestion? Must be break time because you guys are having a hard time with even the easy questions.
Taste?
Yeah, there you go. Taste. There you go. Absolutely. Taste. Obviously not required for digestion to take place, but does it assist in facilitating functions in the digestive system? Yeah, absolutely. Perfect. Excellent. So those are indeed the functions. Obviously, there are accessory structures, but you know what? I think, like I said, I think this is a good stopping point. Let's go ahead and take our next break. And then we'll start talking about some of the accessory structures associated with the oral cavity. So let's leave this here for now. Let's go ahead and take our next break. We'll take a 15-minute break. So we will restart at 10:45. And at this point, I will restart the recording. All right. Any questions before we take our next break? All right. I will see you guys in 15 minutes. Onward and upward. All right, we have talked about the functions and the anatomy of the oral cavity. Let's talk about the accessory structures associated with the oral cavity, starting first with the tongue. What are the functions of the tongue?
Movement, move the food around.
Excellent. All right, so it plays an important role in assisting in the movement of material, movement of food. Presents it to the teeth for that mastication to take place. And again, this doesn't have to be so stinking huge. We also talked about how it initiates swallowing. Excellent. What else? Oops. Taste, as we talked about, it also plays an important role in taste. Again, which I would say is an accessory digestive function. And of course, from a non-digestive functions, what are some of the non-digestive functions that the tongue has? There you go. Laura, you can let people know how you feel about them. What else? Come on, guys. These are the easy questions.
Helping with speech.
There you go. Absolutely. Production of speech. Absolutely. Right. The ability to form the sounds that we make to produce speech is a vital function of the tongue. And if you have siblings, it also allows you to lick things so that you can claim them as yours. Right. So all of those are important functions of the tongue. When we look at the surface of the tongue, it is not uniform in its structure. First, as we already know, because we talked about it in the previous section, there is the posterior third of the tongue, whose anatomy is very different from the anterior 2/3 of the tongue. Notice the posterior 2/3 of the tongue is primarily covered with those lingual tonsils that we talked about in our immune response, that lymphatic tissue that helps us to provide some protection. When we look at the anterior 2/3, though, we see that the anatomy isn't uniform either. In fact, there are 4 main types of finger-like projections or papillae that present themselves on the surface. These are the filiform, the fungiform, the circumvallate, which I think people have gotten tired of saying. And so more and more in the references, you'll just see them as vallate papillae, and the foliate papillae. Let's talk about each one of those. Starting first with the circumvallate papillae. Notice the circumvallate papillae are these very large papillae that are located along the border, basically between the anterior 2/3 and the posterior third of the tongue. Notice behind them, there's a small indentation. That small indentation is known as the terminal sulcus. And again, this is the boundary between the anterior and the posterior part of the tongue. Notice also along the surface, there are these large bumps. These large bumps look like little mushroom caps, and these mushroom caps are what are known as the fungiform papillae. Both the fungiform papillae and circumvallate papillae contain these specialized sensory structures known as taste buds. Taste buds allow us to perceive our sense of taste. And of course, how many taste sensations do we have? Primary taste sensations?
Sweet, sour, salty, bitter.
Umami.
There you go. Sweet, sour, salty, bitter, and umami, or savory. So how many is that? Nobody can count? There you go. Okay, excellent. 5. Excellent. Sorry, I wasn't looking up. 5 taste sensations. Absolutely. We have 5 key taste sensations that are located on the surface of the tongue. And as adults, these are found primarily in our circumvallate papillae and our fungiform papillae. These taste sensations are, of course, chemoreceptors. Right. You may not have thought of it in those terms, but you're absolutely positively aware of it because they basically work as a key in a lock. When something like a sugar comes in, that unlocks the key to turn the sweet sensation, and you taste sweet, right? Lemon tastes sour. Salt is salty. Coffee is bitter. Right, MSG or the juices from red meat, portobello mushrooms, those give that savory sensation of umami. And of course, as you know, because if you've ever gone into a kindergarten class, in that kindergarten class where they're learning about the senses, they have a big picture of a tongue like this. And so we all see that picture and we know that sweet is located right here at the top of the tongue, and bitter is back here in these locations, and salty is along the side, and so on and so forth. We all have this very precise taste map on our tongue. Right? And again, we know that because we go into that kindergarten class and we see that map of the tongue there. But the other place you hear about this precise mapping of the tongue is when you go into Williams-Sonoma. Because when you go into Williams-Sonoma, back in the ancient times when we were allowed to go out and shop, you can buy a single wine glass that costs $35. What makes that single wine glass so special? Well, it is specially formed for the precise wine you like to drink. If you happen to like Merlot, it has the right bulb to it so that when you swirl it, it mixes just the right oxygen with it to maximize the efficiency of the flavor. And not only does it have the right shaped bulb, but the lip is pointed in such a way so that when you pour it into your mouth, it presents the wine to these specific regions of your tongue in just the precise manner to enhance your enjoyment of the wine, making it worth every penny of those $35, right? Yes, no, maybe? Solo cups do just fine? Well, it's a great idea if it actually worked, but here's the problem. Do we all have a precise map to this, to our taste sensations on our tongue?
No.
Well, it's a trick question. Yes, we all absolutely have a precise map, but is all of our precise maps the same? No, that's the key. We all have precise maps, but they're not the same, right? Spring break is right around the corner. And because we're not allowed to go anywhere yet, a perfect thing to do during spring break is to sit around the table with your loved ones, right? And map your taste bud senses, your taste maps. What you do is you take a paper towel, wipe off the surface of your tongue, take a Q-tip and dip it in sugar water, and then slowly paint it over the surface of your tongue. And when you do that, what you'll see is that you only taste sweet in certain locations. So maybe here and here and here and here will be where you taste the sweet. Rinse out your mouth, dry it off with a paper towel. Do the same thing with salt water. When you do with salt water, this will be salty and 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 so on and so forth. You can actually do this, and what you'll see is that everybody in the family has a different taste map. So don't buy those Riedel glasses from Williams-Sonoma. They're BS, right? Red Solo cups work perfectly fine, as Allison pointed out. Excellent. All
I'm
right.
not sure if the filiform papilla is pointing. I don't see a bump or anything.
We'll get there. We'll get there. Absolutely. So we've done fungiform. We've done circumvallate. Notice also on the lateral edges of the tongue are these foliate papillae. They're foliate papillae because notice they kind of like got the ruffled edges like a leaf. They look like foliage. So that is where the name comes from. What is interesting about these foliate papillae is they also contain taste buds, but in infants and early adolescents. In particular, the taste buds found in these foliate region are primarily associated with milk fats and milk sugars. Why might it be useful for these regions to have special taste buds to milk fat and milk sugars in an infant and early adolescent?
So they want breast milk?
Yeah, because their primary food is breast milk, absolutely, in that infant. Absolutely, right? What's interesting about these is that we lose them over time. But this might also explain why some children are highly sensitive to flavors. As I mentioned earlier, there are children who would rather have their left arm ripped off than eat a sprig of broccoli or eat a mushroom or a tomato. Whereas you as an adult, right, those things are pretty mild tastes and certainly not worth having a tantrum over. Well, the reason is that they may be more sensitive to these tastes as adolescents. So one of the reasons that children may be more picky eaters when they're adolescents was one, because they're jerks. But the second reason would be because they may be hypersensitive to tastes because of these foliate papillae. All right. But as Laura pointed out, notice what this picture doesn't show. What this picture doesn't show are the filiform papillae. Although even that's not entirely true, because basically all this smooth surface that you see on the surface of the tongue isn't in fact smooth. This entire surface of the tongue that looks smooth is actually covered by tiny little hair-like papillae. These short, sharp, pointy papillae are what are known as the filiform papillae. Notice if we instead look at it up close, like we see here, we see that what's smooth is really these tall, pointy, sharp-looking filiform papillae. Now, notice with that shape, there's not a lot of room for taste buds. And in fact, they don't provide us any sense of taste. So what do they do for us?
Sensory?
Yeah, they do provide us some sensory information. Think of it this way. If I took a marble, assuming it was clean, and I put it inside your mouth, there certainly wouldn't be any taste to it. But if I put it on the surface of the tongue, would you know where it was? Yeah, you'd feel the tactile sensation. If I had stored it in the freezer before I put it on the tongue, would you feel the temperature change? Or if I warmed it up in warm water before I put it on your tongue, would you be aware of it? Absolutely. And as Ro pointed out, it also provides friction, right? How many people here own or have seen a cat drink water before? How does a cat drink water? I like that, Izzy. What'd you do there? Yeah, basically they use their tongue to basically, it's got that nice roughness on it where basically it has very pronounced filiform papillae, and that friction is able to take advantage of the surface tension of water, and they're basically able to lap it up. Has anyone been licked by a cat before? What's it feel like? Okay, well, Izzy apparently didn't like it, but she's aware it's got that nice, very rough texture to it when that occurs, right? So it's got that very rough texture to their tongue when they lick you, right? When your significant other licks you, it doesn't feel the same. While we have filiform papillae, they're not nearly as pronounced as they are on that cat, right? But we do indeed still have them, and they provide us some, right? And again, think of why. Right? If you think about what that cat eats, the cat is a predator, and typically the things that are being eaten by it don't want to be eaten. So being able to have those pronounced papillae, that extra friction, helps them to not only drink water, but to hold on to their food. Whereas we're omnivores, which means we eat anything we can get through a drive-through. And usually those things don't fight as we're trying to put them down. Right? So we don't need that same roughness to the structure of it instead, but we do still have some friction, tactile sensation, temperature, things along those lines as well. All right, excellent, excellent. So here we see under the light microscopy these different types of papillae. Notice one other thing. Let me see if I can go back. How far back do I have to go? Is it still there? Did I lose the stuff that I was writing? Taste sensations. Apparently. All right. So we had those 5 taste sensations. We'll start here. We'll just erase all that. But notice one of the things we didn't say about those 5 taste sensations is we didn't mention spicy. All right. Sweet, sour, bitter, salty, and umami. But we didn't mention spicy. Why not? Do we have spicy receptors?
They're not on
Spice
the
isn't
tongue.
really a flavor. It's more of like a sensation.
You guys kind of have the right idea. Absolutely. Remember, these are chemoreceptors. They work like a lock and key. The right shaped key turns the lock and you perceive the sensation. Again, you may not have thought of it in those terms, but soda companies, right, like Coca-Cola, like Pepsi, spend gajillions of dollars each year trying to find that perfect chemical that can get in there and turn that lock on your sweet sensations, giving you the perceived sensation of sweet without all those pesky carbohydrates. Right? That is the key to the ultimate diet soda. You get that sweet sensation with no sugars involved, right? Again, it's tricking that chemoreceptor. That's what artificial sweeteners are about. They click that lock and they unlock it. Even though they're not really glucose, they act like glucose. Well, capsaicin, which again is what the typical chemical that is responsible for our spicy sensation, acts like a janitor's key. And what does a janitor's key open?
Everything.
Everything. So when you eat that capsaicin, it turns all the locks on all of the receptors, giving you this massive wave of information. Then that confusing massive wave of information is typically perceived as pain. It also stimulates the temperature receptors on the filiform papillae, tricking you into thinking that it is hot, which is why you sweat as a result of it, why your cheeks become red, right? Why you sweat as a result of that spicy food. It is because you're being tricked into perceiving it as being temperature hot, as well as the painful sensation of that overwhelming sense that we get from that dramatic neural input from that janitor's key turning all of the locks. All right, that is the surface. We talked about this a little bit before. Again, there are many extrinsic and intrinsic muscles. Again, fun with vocabulary. Of course, an intrinsic muscle is a muscle that is completely contained within the tongue. What would intrinsic muscles do for our tongue? All right, we'll start easier. Extrinsic muscles are muscles that start in the tongue and travel out of the tongue, anchoring primarily to what structure? What structure, what bone do most extrinsic muscles attach to?
Mandible?
Mandible.
Not a bad guess, and some of them do, but it's not the primary. If only in 430 we had learned a movable bone that was the primary anchor for the tongue.
Hyoid?
Hyoid bone, there you go, absolutely. All right. Come on, you guys. As our bodies go bigger, our minds grow flowers. Remember, it's great to learn because knowledge is power. So that hyoid bone, right? We got to remember the things we learned in 430 as the movable base. Extrinsic, attaching to that anchor point, is what moves our tongue through space, whereas an intrinsic muscle changes the shape of the tongue. So that extrinsic muscle allows you to stick your tongue out, up, down, left, or out, all around. The intrinsic muscles is what allows you to roll your tongue, right? Curve your tongue, ruffle the edges of your tongue. Right? Move the tip around, things like that. And of course, what controls both the extrinsic and intrinsic muscles of the tongue? I'll narrow it down to 12 things for you. It's one of your cranial nerves.
Was it so it was hypoglossal? Was it cranial nerve 12?
Bingo. Cranial nerve 12, the hypoglossal nerve. Exactly. Excellent, excellent. All right. Anybody remember the nerves involved in taste?
Glossopharyngeal?
There you go. Excellent. So cranial nerve 9, which is, again, gives you more pH, chemical sensation, things along those lines. But the sweet, the salty, the umami, anybody remember which cranial nerve is involved in that? Taste from the anterior 2/3 of the tongue. 7, which was which one again? To touch and feel. Facial. There we go. Excellent. I know I didn't teach you that mnemonic. I don't care. That one's mine. And it's in C7T, so you're not allowed to know that one. All right, excellent. You want to know that one? Go to grad school. Here's more pretty pictures, or just search the internet. Here, again, we see some microscopy of these. And again, notice we can see some of the different types. Here, notice we have those filiform papillae that are, again, you can see how they're narrow and pointy and stick up from that. This is different from our fungiform papillae. Notice this fungiform papillae has that nice mushroom cap. And here we see several of them here. Whereas this big, huge, massively large is that circumvallate. Now, I want you to notice one other thing on these as well. Notice on the fungiform, on the circumvallate, on the lateral aspect, there are these circular structures. These circular structures on the lateral aspects are the taste buds. So notice there's one. And here's one. There's lots of them here. I'm just, so that my circles don't get too confused, I'm just finding individual ones. But notice they go all along the entire length of the circumvallate. Those are these bulb-like structures with a little opening where that chemical goes in and turns that key. And honestly, I don't remember if I have an up-close view of that. Oh yeah, I do. Perfect, excellent. Here we see some more fungiform papillae, and here more clearly, we can see those taste buds along the lateral aspect. All right, so here we see these taste buds here. Here we see the taste buds here. And of course, the surface of the tongue is that non-keratinized stratified squamous epithelial tissue. Here we see the mucosa with some large mucus glands and things along those lines in it as well. Excellent. All right, we did the sulcus terminalis. We have a third frenulum. The third frenulum, the same way that the labial frenella helped to stabilize and anchor the lips, the labia, notice we have a lingual frenulum. And this lingual frenulum is a fold in the mucous membrane that helps to stabilize the tongue, holding the tongue in place. Of course, unfortunately, in some individuals, this lingual frenulum can be too short. It is a term we commonly refer to as being tongue-tied. Of course, you know you're not going to get away with a term like that. The appropriate anatomical term for a too short lingual frenulum is ankylioglossia. But what's the problem if you are tongue-tied? Why is it a big deal if your lingual frenulum is too short?
You can't latch.
Exactly, and that's the biggest issue with it, absolutely. It can affect speech. It can affect chewing and swallowing. Speech is, a big one. But again, most kids don't start talking, till 6 months, 8 months, 12 months, whatever, somewhere around those lines. However, if it is too short, they may have difficulty latching onto the breast and getting the proper vacuum to be able to express the milk. Ankylioglossia, yeah, that's one of the problems with having ankylioglossia is you can't say ankylioglossia, which is somewhat ironic. And of course, how do they resolve that if you have ankylioglossia?
They cut it.
Exactly. They cut it. Absolutely. They cut it. Although I was actually just talking with my dental hygienist, and she was saying actually one of the things they'll go do now is they'll actually laser it. Rather than cutting it, which they used to do in the older days, or remove it to minimize the scarring. She says they actually can laser it to shorten it. I mean, to cut it basically with lasers and basically cauterizes as it goes. It increases the ability to move the tongue in that fashion. So, yeah. So there you go. Speculation. Gene Simmons from KISS tore his lingual frenulum, which is why he's able to extend his tongue as far as he's able to do. Again, back in ancient times, there was a band called KISS. I know most of you don't appreciate that, but that's okay. And as we talked about, again, it produces mucus, it produces that lingual lipase, it does things like that. All right, how are we? Doing good. Excellent. The second major structure, or structures plural, that we are going to be responsible for in the oral cavity or associated with the oral cavity are the salivary glands. Notice there are actually 6 salivary glands. And basically 3 pairs. These pairs are different, obviously, in their location. Let's actually do this. And the composition of the saliva they produce. Lastly, being all exocrine glands, they have different locations for their ducts as well. Now, being exocrine glands, as I'm sure you remember from 430, being exocrine glands, we are going to need to not only know the substance they produce, but we also need to know their structural classification. And we need to know their functional classification. Now, when I say structural classification, what does that mean? All right, what about functional classification? When I say functional classification, what does that mean?
Well, essentially what they do.
Yeah, exactly. How they release their secretion. Anyone remember how many functional classifications there are for exocrine glands? Anyone remember one of them? Yeah, excellent. There are 3, right? Merocrine, holocrine, and epocrine. But remember, epocrine is not a mode of secretion that is used in humans. So that means I'm sorry?
Is that the dog one?
Yeah, it's the dog one, absolutely, right? So it's either going to be holocrine or merocrine, so that makes that easy that way. And remember, structural classification is basically how it's made. How many ducts it has, how many secretory structures it has, and what the shape of those secretory structures are. And since that is all stuff we learned in 430, it's definitely stuff we need to know about these glands in 431. So let's take a look at them. Starting first with our largest salivary gland, and that is the parotid salivary gland. Not only is it the largest of our glands, but it is also the most superficial salivary gland. Notice it basically sits underneath your zygomatic arch, right in front of your ear, sitting on top of the masseter and the temporalis. So basically located in this location in both of those. Notice it has a single very long duct that has to travel over the masseter, over the temporalis, and actually through the buccinator, where it basically releases its content into the vestibule, right? That space between your teeth and your tongue, I mean, your teeth and your cheek, right by the second molar, the superior second molar. Now, let's talk quickly about saliva. What is the function of saliva? Or functions?
Break down food.
Excellent. It's going to contain enzymes that help to break down food. What else?
It softens the food to make it easier for it to digest.
Excellent. It is going to, well, so enzymes to break down the food, it moistens the food, make it easier to digest. Absolutely. Excellent, right? It's very watery that way. Remember, we also talked about how it provides some protection. It's going to contain antibodies like our immunoglobulin A's, remember, that are released in our secretion, some defensins, some things along those lines, making it very, very watery. But notice it isn't just watery. And you know that because, especially if you're an older sibling, you know that you're required by law to kneel on the chest of your younger siblings and dribble just the tiniest bit of saliva out till it almost touches their nose, and then you're able to suck it back up. Because while it produces a very watery component, and that watery component is serous fluid, that does all of these functions we've talked about, it also contains mucin. And mucin, remember, is a special protein that when hydrated with water, what does mucin become again?
Like a lubricant for the mouth.
Right, and what do we call that lubricant of the mouth that mucin becomes?
Mucus.
Mucus, there you go. It becomes mucus. So notice there's basically two main components to saliva, mucus and serous fluid. Now, let's think about this. This big, huge, massive gland has one duct and one duct only that comes out of it. Lots of ducts inside of it, but only one that comes out of it. And it travels a long distance to get to its destination. Based on that, since we know saliva can either be watery or have mucus in it, would you expect the saliva produced by the parotid gland to be very mucousy, to be thick and viscous like that mucus would be? Would that easily travel a long distance in a single duct? No. So not only is this the largest salivary gland, but it mostly produces serous fluid. So we have this parotid gland, again, inferior to the zygomatic arch, primarily produces serous secretions. Again, things like the salivary amylase, things like the defensive, things like the antibodies, all of those types of things. and a single duct that travels all the way to the vestibule adjacent to the second superior molar. Now, knowing all of these things about our parotid gland is going to help us to be able to recognize it when we look at it histologically. Notice here we see an exocrine gland, and I'm sure as we remember, and let's do this because I think it'll make things easier. I'm going to cheat and steal a corner here. There we go. Like all exocrine glands, it is comprised, and I think I may need to make this shorter to fit it all in here. It is comprised of secretory structures and ducts. I'm sure all of you remember this from 430, but humor me as I go through it again. What do the secretory structures do?
Secrete.
Yeah, they make the secretion. What do the ducts do again?
Transport.
Oops. Secretory structures and ducts. That's what I meant to say. Excellent. Absolutely. Their sole purpose is transportation. They're conduits from getting from one location to another. So all they do is they're the PVC pipe that carries the secretion out of the body. They don't modify it. They don't make it. They don't change it. They don't do anything with it at all. And so all exocrine glands have that. And so notice as we look at this slide histologically, we can see here these tubular structures, which are the ducts, and pretty much everything else, which are the secretory structures. So we see both parts that make up an exocrine gland. We see the ducts, we see the secretory structures. Now, I have a vague recollection of 430 that we learned that if a gland is made up of one duct and one secretory structure, we called it a simple gland. Do we just have one secretory structure here? Do we just have one duct here? No, we have lots. So we know this isn't simple. I also remember if I had one duct and two or more secretory structures, we would call that a branched gland. But I see more than one duct. I definitely see more than one secretory structure, but I also see more than one duct. So that can't be it. So I know when a gland is made of two or more ducts and two or more secretory structures. What kind of gland is that again? Anyone remember? Someone said something, but you're quiet. I couldn't quite hear you. I'm sorry, I still couldn't hear you. You're too quiet. Are you talking to us or are we just overhearing someone's conversation? All right, well, for those of you who don't remember, it is compound. There we go. Excellent. Compound. Excellent. So clearly this is a compound gland. Okay, that helps. Remember also, there are basically two shapes to the secretory structures. And you remember what the two shapes to a secretory structures were?
Tube and
Yep.
alveolar.
Tube and ball shape. Excellent. Tube-shaped glands were called tubular. That makes it easier. So ball-shaped glands were baller.
Alveolar.
Yeah. Alveolar, or remember, you could also use the term acinar. Excellent. Perfect. So as I look at these secretory structures, are they more tightly compact ball-shaped secretory structures, or am I seeing tubes at all sorts of different orientations?
Different orientation.
Well, I think to me, again, while they're all not perfect circles, they're all pretty compact circular shaped structures. So it turns out this particular gland happens to be, and since I've already written out our rules, I can go ahead and erase it. Whoops, no, didn't want to erase that. I wanted to erase that. Nope, stop it. There we go. Structurally, this is a compound alveolar gland. All right, it's a compound alveolar gland. Now, these alveoli, what did we say they make? Do they make the mucin or do they make the watery serous fluid? What's likely to come out of that parotid gland?
Serous fluid?
Serous fluid.
Serous fluid. So we could call them serous acini or serous alveoli. Yeah, I'll use the same terms because they make serous fluid. Dark pigmented structures make serous fluid. So excellent. And so then the last thing we have to figure out is what its functional classification is. Holocrine is where the cell fills itself up, explodes, and releases a very viscous, very organic type of material. Whereas merocrine, remember, was where they release things by exocytosis. Which way is this most likely going to release its serous fluid?
Exocytosis.
Exocytosis. So it uses the merocrine mode of secretion. So all we see are serous alveoli, many of them, and multiple ducts. That tells us this is a compound alveolar gland, means it's the parotid gland that produces primarily serous fluid, and it releases it using the merocrine mode of secretion. So not only do we now know everything that the parotid gland does, but we can recognize it histologically as well. All right, questions on that? Let's do it again. Let's talk about our sublingual gland. Let's take a look at our sublingual gland. Notice our sublingual gland is this one here, located under the tongue. That's convenient based on its name. Again, paired on both sides. But notice something interesting about it. This doesn't just have one terminal duct. It has many, whoops, hold on. Many terminal ducts that are carrying secretions out from it. Notice it has many ducts and they're very short in length. With this type of anatomy, do you think this is going to be more useful for really watery secretions, or do you think that we might, for something thick and viscous and like mucus, that might be more convenient to have multiple very short ducts? Yeah, so notice this one is going to primarily produce mucus. Oops. Or again, if we want to be more specific, mucin, which is a protein that is released and hydrates to become mucus. So it's on the floor under the mouth, and it's mucus secretions release out. It also produces some buffer, provides some lubrication, and greases the wheels for that food as it is being swallowed down. Notice it has multiple ducts that exit along the lateral aspect of the tongue. I think I have a picture that we're going to get to that shows it, but notice if we go back to this picture of the tongue, notice these right here along that lateral fold of the tongue, those small openings are the ducts for the sublingual glands. Notice not the ones on the midline. That's something different. We'll talk about that in a minute. But the multiple lateral ones, those are the openings for our. Oh, I did have a picture. See, there you go. Even more clever than I thought I was. Perfect. All right. Now, we know this. Let's look at this one histologically. Notice when we look at this, this looks very, very different from what we saw before. Notice we are still seeing ducts. Here is a duct, and here is a duct, and here is a duct. The fact that it has multiple ducts right off the bat should warn you that this is a compound gland. So again, we're looking at sublingual, right? If we have two or more ducts, we know this is going to be a compound gland. However, now let's look closer at the secretory structures. Notice when we look at the secretory structures, some of these look more circular in their shape. Some of them are more oval in their shape. Some of them are more elongated in their shape. All right, so we have some oval, we have some circles, we have some elongated. Notice it's almost like you had a tubular structure that you were cutting at different angles. And as you cut them at different angles, these would have very distinctly different shapes. But notice the other big, huge difference when we look at these secretory structures. Notice these secretory structures are very clear in color. Not quite adipose clear, but very clear in color. Again, remember, color is one of those artifacts we don't want to spend too much attention to unless it's important. And here it's important. What makes these secretory structures so clear? Mucin. Remember, mucin is a protein that a lot of stains don't bind to. So most stains, when we put it on the tissue, it doesn't bind to mucin. So mucin looks very, very clear. So notice how with our previous salivary gland, all the secretory structures were very dark. Here, the secretory structures are very clear, and they're much more irregular in the shape. So notice here, our secretory structures are going to be tubules, and they're going to contain mucin. So we could even call them mucus tubules. And of course, because we have many of them, structurally, it is a compound tubular gland. So that is its structural classification, compound tubular gland, and we recognize that from the tubules. Any idea its functional classification? It's making and releasing a lot of protein, mucin. Is that something it's going to want to burst and release? No, so it's also going to use the merocrine mode of secretion. Remember, we said merocrine was the most common type. So when all else fails, guess merocrine. So this one produces mucin, mucus. It's a compound tubular gland, that uses the Merocrine mode of secretion. And again, I can't emphasize this enough. Look at the difference between the histology of this gland and the histology of this one. Both have ducts, both have secretory structures, but the secretory structures are very, very different. Dark, small, circular, compact. Dark because they're making serous fluid, small and compact because they're alveoli. Bigger, lighter, more irregular in shape because this one has mucus tubules. And let's look at our third gland. We'll talk about the wording of it in just a second. But here's what this one looks like histologically. Notice again, the easiest thing to find is the ducts. Here is a nice big duct right here. In fact, it's actually two ducts side by side. That's easy to see. But notice when I look at my secretory structures, is there just one type of secretory structure here? No, notice I have nice, tight, compact ball shape that are very, very dark. What would these dark ball shaped things be?
Alveolar.
Yeah, these are serous alveoli. Excellent. But notice I also have, oops, no, hold on. You need to be green. Some things that are very light in color, some of which are circular, some of which have a very elongated shape to them, right? More oblique and turn to them. And what are these things?
Mucus tubules.
This is our third salivary gland. Our third salivary gland is the submandibular. It's not the largest, but it makes the majority of our saliva. All right. Now, when we think of the composition of the saliva that it's producing here, is it serous fluid? Is it mucin?
Both.
Both. Making both serous fluid and mucin. So it's mixing the two together. So what would its structural classification be, do you think? It has two or more ducts, so it's compound. So is it compound tubular or compound alveolar?
Both?
Both.
Tubular alveolar.
There you go. And in fact, if you remember back to 430, we have a name for a type that has both. We call it a compound tubulo. Oops, tubulo alveolar. And so this is a compound tubulo-alveolar gland that produces both serous fluid and mucin. And guess what its functional classification is? There you go. Merocrine. Because after all, if the tubes use merocrine and the alveoli use merocrine, then when you put the two together, they use the merocrine mode of secretion. So notice, just from looking at these histologically and paying attention to the secretory structures, histologically, you can tell the three salivary glands apart and also tell what they do. So let's take a look at that submandibular gland. Submandibular gland is the most inferior of all the salivary glands, located way down here in the depths of the mandible, again, on both sides. Notice it has a single duct, but its single duct is relatively large and medium in length, which again makes sense because it's large and relative and medium in length because it's going to have some mucus in it, but it also has some serous fluid in it as well. And notice this is the one that releases its materials right next to the lingual frenulum. Again, you may not in your entire life have ever thought of this before, but you, or I'm guessing someone you know, at one point in your life or another has been able to express saliva projectilely out of these two ducts located right next to the lingual frenulum, right? Spraying you or somebody else you love with them. What do they call that?
Gleeking.
There you go.
Gleeking.
I don't know if there's a technical term for that, but that's the term I've always heard for it. Absolutely. That ability to do that is that expression of, and I'm not able to do it, otherwise I'd demonstrate for you. Maybe I wouldn't, I don't know, but I can't, so it doesn't matter. That ability to express that saliva out of those ducts from the submandibular gland. So these are the two ducts right next to the lingual frenulum for that mucus producing, both serous fluid and mucus producing submandibular gland. So there you go. Just that easily, we have identified our 3 salivary glands on an illustration like this. And notice also another place where you see this really, really nicely. If we go back to here, this is that, again, the CRC site. Notice again, here we see it really nicely as well, where again, notice here is that parotid gland, and we see its duct and how it's coming out. Notice it's coming out into the superior vestibule next to the second molar. Here we see the sublingual gland and its ducts coming up into those little openings that they've made right there. And then I guess I'll change colors to make this a little easier. There's our submandibular, and notice we can see its duct coming out, kind of hidden between here, and then coming out to the front of the oral cavity there as well. So there's lots of great places where we can see this very, very clearly. These 3 glands, paired glands on the lateral aspect of the adjacent to the oral cavity. They're ducts, and so we know their locations, we know their names, we know the chemical composition of the saliva that they produce, and we can recognize them histologically, which means we also know their structural classification and functional classification. And I do fully expect you to be able to recognize them from a chart or a model or some structure like that, as well as being able to recognize them histologically and being able to recognize what gland and what each of these components produce. All right, questions on that? All right, collectively, these 3 glands, and again, this is an average. It depends tremendously on 3 factors: how often you eat, the composition of the food you eat, and the volume of the food you eat. Those 3 factors can modify all of these significantly, but on average, a person produces about 1 to 1.5 liters of saliva during the course of the day. And again, remember the majority of that, close to 70%, comes primarily from the submandibular gland, which again is not the largest. The largest is the parotid, but it is the one that produces the most secretion. It's mostly water, but like we said, a lot of other stuff. Electrolytes are immunoglobulin A antibodies that, again, remember the dimers that provide that binding of pathogens before they can get in. Lysozymes, which are antibacterial in component, defenses, other enzymes, mucin, buffers. Right? And again, the goal of this is to mix with the food. Right? Again, fancy vocabulary terms. Our food does not enter the alimentary canal and look the same way when it exits. Food basically undergoes 3 iterations on its mystical, magical journey through the alimentary canal. And that first, on its mystical, magical journey, it enters as a cheeseburger, but after you have masticated it and mixed it with your saliva, right? Do you necessarily want your friend to spit it back out so that you can have a bite? No, the composition of it is changed dramatically to form a substance we call a bolus. So here in the mouth, with mastication, with saliva, that cheeseburger has undergone its first iteration, its first change, and has become bolus. And then our job is to then present that bolus to the posterior part of the oral cavity so it can continue its mystical, magical journey along the way. All right, so again, you put that cheeseburger in, you take a bite of that cheeseburger, you chew it the 35 times you're supposed to before you swallow it, mixing it with all the saliva, mixing it with all the mucus, mixing it with all the serous fluid to form that bolus. And from there, it is ready to continue on its mystical and magical journey. And does anybody remember the primary nerves responsible for controlling the salivary glands? Again, primarily under parasympathetic control, but again, our sympathetic will as well. But anybody remember which cranial nerves are primarily responsible for controlling our salivary glands?
Glossopharyngeal.
Yep. Glossopharyngeal, 9, and facial, 7. There you go. There you go, 7 and 9. Via, remember, our submandibular ganglion and our otic ganglion, 2 of only the 4 terminal ganglia in the parasympathetic nervous system that are named. I know none of you remember that, I don't care. But it's cranial nerve 7 and 9. Excellent. The last accessory structure we need to be able to identify associated with the oral cavity are indeed the teeth. I don't want to spend too much time on this. Again, you're going to need to work on the anatomy. So again, the things you need to absolutely positively know is the anatomy. Remember, tooth is and again, you have your anatomy list that you're responsible for. So I'm not going to go over all of this, but there are some key components I want to remind you of. A tooth is a bone-like structure, but is not a bone. It does have similar composition to bone in that on the outer exposed surface of it, we have a substance called enamel. And like bones, that enamel is primarily made of those hydroxy apatite crystals. Remember, hydroxyapatite crystals are the strongest substance made by the body. However, remember the big difference between the enamel of your teeth and your bones is that the enamel of your teeth does not contain collagen fibers. Remember that collagen fibers gives your bone some flexibility, gives some give. Hydroxyapatite crystals are very strong, but they don't have any give. And so that makes them rigid and therefore potentially brittle, right? If you, a couple months ago, right, were cheering on the fact that Tom Brady cheated his way to another Super Bowl win, and so you're having a big old bowl of New England clam chowder to celebrate that, and you bit on that clamshell, right? Is there any give or flexibility in your enamel when you bite down on that clamshell? No,
Nope.
So you can crack that enamel as a result of that. The internal substance of the tooth is also bone-like, but not truly bone, in that we have these, as you can see, these cylinders of essentially what is very similar to a calcified cartilage. It is similar to a calcified cartilage, but not exactly calcified cartilage. And this substance is known as dentin. Also, like a bone, the inside is actually hollow. Of course, the inside of the bone, a long bone in particular, we call the medullary cavity, but this is the pulp cavity. And while the medullary cavity contains bone marrow, what does our pulp cavity contain?
Blood vessels.
Nerves.
Yeah, blood vessels and nerve and connective tissue, which all collectively is known as
Pulp.
pulp. There you go. The pulp cavity contains pulp. See how convenient that is? There you go. Excellent. Right? Notice, obviously, there needs to be a way for these blood vessels and nerves to get in. So notice, because the tooth is somewhat cone shape, it has an apex at top, it has a base near the top, apex at the bottom, base at the top. There's the apical foramen, which is the entryway for those blood vessels and nerves to get in. As you also know, if the enamel is damaged, you can get a cavity, and that cavity can cause pain. And so what does the doctor do in that instance?
Either filling or root canal.
Yeah, well, if it doesn't go down to the nerve, they can fill it to protect it. But if it goes down to the nerve where it's causing injury or pain, he basically takes the drill and hollows the space out, essentially killing the tooth and then filling it. But again, stopping the pain, and that we call a root canal. Absolutely. All right. Excellent. Clear my drawings. The tooth is truly not a bone, but it is bone-like enough where the connection between either the mandible or the maxilla and the tooth is considered a joint, which is awesome. Because in 430, we learned that when talking about joints, we need to know the structural classification of our joints. We also know that all structural classifications have specific types of joints. And we know that also all joints have a functional classification. And conveniently for us, I'm sure you guys remember all of that. So let's start easy. What is the structural classification of the joint of the tooth and either the mandible and the maxilla?
Anasmorph.
All right, I like where you're going. That is definitely joint related. But remember, let's think of it this way. When we talk about the structural classification, remember we're talking about what holds the two bones together. And like
Fibers.
any good student, I've got the picture here in front of me, so I'm going to cheat. I see that here is the bone-like structure that is the tooth. Here is the bone that is the mandible. And notice in between them, I see that there is a periodontal ligament, a fibrous connective tissue that is holding these two bone-like structures together. So what would the structural classification of this joint be?
Fibrous joint.
Fibrous joint, right? Because it is fibers that hold them together. Excellent. Let's go to the next easy one. How much wobble should your teeth have in the joint of your jaw?
None.
None. So it should not allow any movement. So what would the functional classification of a joint that did not allow any movement to be? I think Izzy's got the right answer there. It is a synarthrosis. So then that just leaves us to the specific type. Remember, there are three types of fibrous joints. The sutures of our skull was one of them. Our syndesmosis, like where a ligament, a membrane, holds the two together. but the specific joint that holds the teeth in place, if I remember correctly, it was my favorite because it's the most fun to say. What was the specific type of fibrous joint that we called that joint between the tooth and the jaw?
Was it
Gomp from something with a G.
There you go. Gomphosis, which allows you to chew gum. See how easy that was? So now for our tooth, we remember our structural classification, our specific type of joint, and our functional classification, which absolutely positively is testable material on the exam. Now, let's do a little bit more anatomy. I need to erase these pictures over here so we can get a better look. Notice, as we talked about, down here where the tooth is embedded in the bone, as we talked about, we have the pocket, the alveoli, or the hole for the tooth that it sits in. And we have the tooth itself. And as we talked about, we have that periodontal ligament that holds it together. But notice this periodontal ligament attaches not to the dentin, but if you look very closely, you see that there is a thin layer of material on the outer surface of the bone. Notice we see it nicer when we look down here. The enamel is big and bright and white, and that is the exposed part. But notice the rest of the tooth is covered with this tan material. This tan material is called cementum. So technically, our gomphosis is a joint between the cementum of the tooth and the alveoli or apical, no, pardon me, not the alveoli, the pocket in the tooth and that periodontal ligament. This part that is embedded in the bone is known as the root. Notice the part on top that is covered with enamel is known as the crown. And notice right here in the middle is a narrow region known as the neck. Notice the neck of the tooth isn't covered with enamel. It's also not protected by the bone. So notice in this location, the cementum is only protected by the gingiva, the gums. It's one of the reasons why it's so important to floss, because if you don't floss properly, what can actually happen is your gums will start to recess, they'll start to retract, and as they retract, they expose that neck region of the tooth, which isn't protected by enamel and isn't protected by bone, and you're much more likely to get cavities in actually this location than you are actually in the enamel of the teeth. So that gum hygiene is very, very important for that region. So again, know the anatomy, know the composition. And then lastly, teeth come in different flavors. There are the incisors, the canines, the bicuspids, or premolars, the molars, the cuspids, bicuspids, tricuspids, cuspids, incisors, canines, premolars, molars. Know the functions of the four types. Know that we come in two different versions. We have our milk teeth, our baby teeth, our deciduous teeth, and our permanent teeth. Know the numbers and know our dental formula. The dental formula basically is the composition of teeth on one quadrant of the oral cavity. So notice for an adult, it is 2, 1, 2, 3. We have 2 incisors, 1 canine, 2 premolars, 3 molars. Lastly, your book does a nice job of saying when exactly the teeth come in. I will say that the timing of when the teeth come in varies dramatically from person to person, and there's a very strong genetic component to that. However, what I would say is for the most part, for most individuals between birth and 2 years of age is when most of your deciduous teeth have come in. And then starting somewhere between 7 and the ripe old age of 18 to 25, when your wisdom teeth come in, because you're old and wise at that point, is when your permanent teeth, your adult teeth, start to come in. So you don't need to know the exact timing, but know the relative ages of when these are coming in. And obviously the difference in numbers between the two. All right. So again, we did all of that, we did all of that, we did all of that. I didn't want to spend too much time on this. Pretty pictures. Did that, did that, did that, did that. Okay, again, I don't want to spend too much time. So there you go, 6 to 24. It's all here in the lecture notes. It's all here in your textbook. Again, I want you to be comfortable and familiar with that, but we still have a lot of material to get to. So the last thing that I will talk about for today is what happens when the food leaves the oral cavity. When the food leaves the oral cavity, and we'll talk about the process of that. We'll actually save that for the beginning of our next class. But it enters into the muscular tube that we commonly think of as the throat and is appropriately known as the pharynx. The pharynx is the muscular tube that we commonly think of as the throat. And notice it's the posterior part of all of these cavities. In fact, that's how the 3 different regions get its name. There's a portion of the pharynx that is the posterior entrance for the nasal cavity, and it is known as the nasopharynx. This is obviously where air comes in. The oral cavity posteriorly opens into the oropharynx, which is where our food and drink comes in. And as we'll talk about in our next lecture, when we talk about deglutition, when we talk about swallowing, we need a regulatory structure here that is going to help us to send air in one direction and food and drink into another. And that specialized structure is known as the larynx. So the portion of the pharynx that is open to the larynx is the laryngopharynx. And then inferiorly, the pharynx feeds into the esophagus. So here we see this with the pretty picture here. Here's another pretty picture that again shows these openings, the 3 regions to the pharynx and how inferiorly it leads into the esophagus. And like I said, what we will talk about at the beginning of our next class, because it's noon, so I think this is as far as I wanted to go for today and where I'd hoped to get to for today, is we will talk about this process of deglutition, the process of swallowing. We've done everything we need to do with the oral cavity, and so now we get to continue on our merry way into the pharynx and into the esophagus. All right, now there is one more thing I want to talk about for the pharynx. When we talked about the oral cavity, notice we mentioned about how 5 of the 6 processes of digestion occur in the oral cavity. How many of our digestive functions occur in the pharynx?
Just movement?
Yeah. One function only. What is it? Movement, propulsion. In fact, for both the pharynx and the esophagus, as we'll talk about the esophagus in the next class, basically these muscular tubes are essentially just conduits, passageways, hallways, PVC pipe. Right? The PVC pipe carries the water from the spigot to your sprinkler heads. It doesn't modify it. It doesn't change it. It doesn't make it. It doesn't do anything other than carrying it from one location to another. And so whereas the oral cavity does pretty much everything, the pharynx and the esophagus pretty much just do one thing: movement, propulsion. The job of them is solely to get the food from the oral cavity into the stomach, where the next big major processing is going to take place. And so that's our goal in our next class. The beginning of next class, we'll talk about this process of deglutition, where we move the food from the oral cavity into the pharynx, into the esophagus, so that it can reach the stomach and more fun things can occur. All right. Questions on any of that? All right, excellent. Again, as I warned you, this is usually 3.5 lectures worth of material. We don't have any labs, so really it's just going to be a lot of talking for these, but we're going to try to cram it into 3 days. So we got a fair amount to cover, but like I said, we got as far as I'd hoped to get today, which is exactly where I wanted to get, finished with the oral cavity and on our merry way from there. And so we are on schedule and should be good that way. All right, questions on any of that? All right, you guys have an excellent day, and I will see you on Wednesday.